A snake vanishes beneath the surface and does not come back for air. Minutes pass, then more. In water that holds no breathable oxygen for a reptile, its body remains submerged, weaving through coral corridors as if suffocation were a myth. Any land snake forced below the surface this long would drown. Yet this one continues to hunt, heart beating slower, lungs held full, skin quietly stealing life from the sea itself. The reef around it pulses with movement. Shafts of filtered light cut down through blue water, illuminating labyrinths of branching coral and dark overhangs where fish cluster in nervous schools. The sea snake moves without hurry. Its body is laterally compressed, tail flattened like a paddle, every undulation precise and economical. It is not built for sprinting. It is built for endurance. Each motion is measured because every flick of muscle burns oxygen, and oxygen is the one resource this hunter cannot replenish at will. Inside its chest, a single elongated lung runs nearly the entire length of its body, functioning not only as a respiratory organ, but as a buoyancy regulator. Before diving, the snake surfaces briefly, taking a deliberate breath. That lung inflates like a slender internal balloon, storing air that must last until the next ascent. But the breath is only part of the strategy. Once submerged, something subtler begins. Water presses against its scales, flowing across skin that appears impermeable at first glance. Yet beneath that patterned exterior lies a network of capillaries unusually close to the surface. In certain species of sea snake, particularly those that spend most of their lives offshore, the skin itself participates in respiration. Oxygen dissolved in seawater diffuses directly into the bloodstream, supplementing what was stored in the lung. It is not enough to replace a breath entirely, but it can extend a dive by a critical margin. In still water, the effect is modest. In moving currents that refresh the boundary layer around the body, it becomes significant. The physics is unforgiving. Oxygen concentration in seawater is far lower than in air, and diffusion across skin is slow compared to inhalation through lungs. But slow is acceptable when metabolism is suppressed. And here lies the second adaptation, the deliberate throttling of life's pace. Once submerged, the sea snake's heart rate drops dramatically. Blood is shunted strategically, favoring brain and essential organs while limiting flow to tissues that can tolerate temporary hypoxia. Muscle activity decreases unless required for hunting. Even digestion slows if prey has recently been consumed. The body becomes a machine operating in low-power mode. This metabolic suppression is not accidental. It is evolution's answer to constraint. A reptile cannot extract oxygen from water using gills. It must bring its air supply with it. The only way to stretch that supply is to burn it more slowly. In laboratory studies, some sea snakes demonstrate dive times exceeding 2 hours under controlled conditions. In the wild, most dives are shorter, often lasting between 30 minutes and an hour. But during extended foraging in deep reefs or open water, the difference between a short dive and a long one can mean the difference between feeding and starvation. Hunger tightens this balance. The reef does not surrender prey easily. Many sea snakes specialize in hunting small fish hiding in crevices or eels that retreat into narrow coral tunnels. To capture them, the snake must probe confined spaces, writhing into cavities where turning around becomes difficult. Each probing movement costs oxygen. Each failed strike drains reserves that cannot be replaced underwater. The predator must calculate instinctively. How long can I remain below before ascent becomes mandatory? Yet the sea snake's design pushes it toward risk. Its venom is potent, often neurotoxic, capable of immobilizing prey rapidly. This allows the snake to conserve energy during subduing. Rather than wrestling or constricting, it delivers a precise bite and waits. Paralysis spreads quickly through small fish, reducing struggle and oxygen expenditure. The efficiency of its venom is not merely a weapon, it is an oxygen-saving device. But even venom cannot shorten every hunt. Some prey wedge themselves deep within reef matrices, forcing the snake to linger, head inserted into darkness, body coiled outside in open water. In these moments, The snake's entire physiology hangs on a fragile equation. Its lung volume determines buoyancy. Too much air and it floats upward. Too little and it sinks uncontrollably. Muscular effort to maintain position increases oxygen consumption. A slight miscalculation and the need to surface may interrupt the hunt at the worst possible instant. The ocean compounds the challenge. Currents sweep across reefs unpredictably, increasing the flow of oxygenated water across the snake's skin, enhancing cutaneous absorption, yet simultaneously demanding more muscular correction to hold position. In calm lagoons, oxygen uptake through skin is limited by stagnant boundary layers, but energy costs remain low. In turbulent passes, oxygen diffusion improves, but so does metabolic demand. There is no simple advantage. Every environment forces a recalibration. Then there is depth. As the snake descends, pressure compresses the lung, reducing its volume and altering buoyancy. The snake compensates with subtle body adjustments, sometimes releasing small amounts of air before diving to achieve neutral buoyancy at a target depth. But lung compression also reduces the surface area available for gas exchange once it surfaces. A rapid ascent after prolonged submersion can lead to abrupt changes in blood oxygen levels, demanding precise physiological control to avoid disorientation. The deeper paradox emerges when considering carbon dioxide. Oxygen depletion is not the only constraint. Accumulation of carbon dioxide in blood triggers the urge to breathe. Sea snakes exhibit a remarkable tolerance for elevated carbon dioxide levels, allowing them to delay resurfacing beyond what many terrestrial reptiles could endure. Their chemoreceptors are less sensitive, or their buffering systems more robust, enabling them to operate closer to physiological limits. But this tolerance is not infinite. Push too far, and muscle coordination falters, reflexes slow, and vulnerability increases. Predation risk adds another layer of tension. While hunting, sea snakes are not invincible. Large fish, sharks, and even some birds can prey upon them. Surfacing frequently exposes them to aerial predators. Staying submerged too long risks hypoxia. The snake's behavior becomes a compromise between two dangers: suffocation below and exposure above. It must choose the lesser threat, moment by moment. Reproduction complicates the oxygen equation further. Many sea snakes are viviparous, giving birth to live young at sea. Pregnant females carry developing embryos within their bodies, increasing metabolic demand. More tissue requires more oxygen, yet the need to feed intensifies during gestation. A gravid female may push dive times closer to her limit to secure sufficient prey, balancing her own survival with that of her offspring. The ocean does not offer maternity leave. It offers only constraint. There is also an overlooked advantage hidden within the lung's elongated structure. Extending nearly the full body length, it functions as a hydrostatic organ, aiding in balance and stability during swimming. This reduces unnecessary muscular adjustments, conserving oxygen over long distances. The snake's entire body is integrated around efficiency. Even its scales are smooth and overlapping in ways that reduce drag, minimizing energy expenditure per unit distance traveled. And yet, despite all these adaptations, sea snakes remain air breathers. They must return to the surface. When they do, the ascent is often abrupt. A sinuous form rises through blue water, breaking the surface with a small ripple. A quick inhalation, sometimes lasting only seconds, refills the lung. Then it disappears again. The surface visit is brief, almost furtive, minimizing exposure. The larger ecological implication of this oxygen balancing act is profound. By exploiting underwater niches for extended periods, sea snakes occupy a role few reptiles could sustain. They regulate populations of small reef fish and eels, shaping micro-communities within coral structures. Their predation patterns influence which species dominate crevices and how energy flows through the reef system. Oxygen limitation does not restrict them to the margins. It sculpts their influence. But the balance is delicate. Warming oceans reduce dissolved oxygen levels in many regions, shrinking the margin available for cutaneous absorption. Hypoxic zones expand in certain coastal waters, making extended dives more dangerous. A system finely tuned to narrow oxygen gradients faces new uncertainty. The The snake's ancient compromise between lung and skin, metabolism and motion, may confront conditions outside its evolutionary experience. In twilight waters, a sea snake glides along a reef wall, its body a ribbon of patterned muscle. It pauses near a shadowed crevice, senses flickering movement, and slips forward. The hunt unfolds slowly, deliberately. Time underwater stretches. Oxygen reserves thin. Yet the snake remains composed, heart slowed, metabolism restrained, skin quietly absorbing what the sea can spare. When it finally ascends, breaking the surface for a swift breath, the motion seems effortless. But beneath that smooth rhythm lies constant negotiation. Every dive is a wager. Every hunt is measured against the invisible countdown of oxygen. Survival here is not about speed or brute force. It is about operating at the edge of suffocation without crossing it. In a world defined by water, this reptile lives between two realms, borrowing air from above and oxygen from below, slowing its own pulse to stretch seconds into minutes. The ocean never stops demanding breath. The sea snake never stops calculating how long it can refuse. An octopus escapes through a hole no wider than its eye, leaving behind a predator that could crush bone but cannot follow through stone. There is no dramatic chase, no burst of speed, no violent struggle, just a quiet, vanishing act. One moment the animal is cornered against a reef wall, skin darkened in alarm, siphon pulsing. The next, its body liquefies into a crack, arms folding and compressing until only a ripple of silt marks where it once existed. In the deep sea and on crowded reefs alike, survival does not belong to the strongest. It belongs to the most adaptable. The seafloor is not a gentle landscape. It is a labyrinth of sharp coral, shifting sand, jagged basalt, and vertical drop-offs that vanish into blue-black emptiness. Pressure increases with depth, light fades, and currents twist unpredictably around rock structures. In such conditions, armor is heavy, rigid bodies are liabilities, and predictable movement is fatal. The octopus has evolved in direct defiance of those constraints. It carries no shell. Its skeleton is absent except for a small internal remnant known as the gladius in some related species. Its body is almost entirely muscle and nerve, bound by collagen and fluid. What looks fragile is in fact strategic. The defining advantage is softness. Without rigid bones limiting form, an octopus can compress its mantle and funnel its arms through gaps astonishingly small. If its beak, the only hard structure in its body, can pass through an opening, the rest can follow. This flexibility is not random collapse. It is coordinated deformation. Circular and longitudinal muscle fibers in the arms act as hydrostatic skeletons, generating stiffness or slackness as needed. By altering internal pressure and muscle tension, the octopus can turn an arm into a rigid probing tool or a supple ribbon that wraps around rock and prey alike. In extreme environments, this capacity becomes more than escape artistry. It is habitat access. Many predators are too large or too rigid to enter the narrowest crevices of reefs or volcanic rubble. The octopus turns these cramped spaces into fortresses. It occupies dens beneath rocks, lining entrances with shells and debris like barricades. These are not accidental piles. Observations show deliberate selection and placement, an act that suggests planning rather than instinctive scattering. A den becomes a base of operations, a vantage point from which the octopus can extend arms into open water while keeping most of its vulnerable body concealed. Yet physical flexibility alone does not explain survival in low-visibility waters. Intelligence fills the gap where brute force fails. The octopus brain is not centralized in the same way as that of vertebrates. A large portion of its neurons reside within its arms, granting semi-autonomous control. Each arm can taste and feel simultaneously, exploring texture and chemistry without direct oversight from the central brain. In murky depths where light scarcely penetrates, this distributed nervous system becomes a sensory network. The animal does not merely see its world, it feels and samples it continuously. Vision, however, remains powerful. Octopus eyes are sophisticated, capable of detecting polarized light and subtle contrast shifts. Even in dim conditions, These visual systems can identify movement patterns that reveal prey or predator. But sight is only part of the equation. The skin itself is a canvas of chromatophores, iridophores, and leucophores that can alter color and texture in fractions of a second. When threatened, the octopus does not simply hide, it transforms. Skin papillae rise to mimic the roughness of rock. Pigment sacs expand or contract, creating patterns that match coral, sand, or shadow. The deception is dynamic, adjusting as the background changes. This camouflage is not a static trick. It is an active negotiation with surroundings. Research has demonstrated that octopuses can adjust patterns even when colorblind, relying on brightness and texture cues to approximate their environment. The brain interprets visual input and translates it into muscular signals, controlling thousands of pigment cells. The result is an animal that can vanish not by fleeing, but by blending. Yet even camouflage is not the pinnacle of octopus strategy. Problem-solving defines its survival in extreme conditions. In laboratory settings, octopuses have been observed unscrewing jar lids to access prey, navigating mazes, and learning through observation. In the wild, these cognitive abilities manifest as opportunistic ingenuity. Coconut shells are collected and carried as portable shelters. Discarded bottles become temporary refuges. This is not instinctive sheltering behavior seen in crustaceans occupying shells. It involves selection, transport, and later assembly, implying foresight. In deeper waters, where pressure increases and light disappears entirely, species such as the Dumbo octopus drift above abyssal plains. Here, adaptability shifts from camouflage to energy efficiency. Movements are slow, deliberate, conserving energy in an environment where food is scarce and oxygen levels may be low. The absence of a rigid skeleton reduces energetic costs of locomotion in buoyant water. Arms ripple like fabric, generating lift without frantic effort. Intelligence here becomes restraint, a refusal to waste resources. Hunting reveals another dimension of flexible strategy. Some octopus species stalk crustaceans by extending an arm into burrows, probing with sensory suckers that detect chemical traces. Others ambush fish by spreading a web between arms and lunging forward in a sudden engulfing motion. The choice of tactic is not uniform. It depends on prey type, environmental structure, and risk assessment. An octopus confronted with a venomous lionfish may deploy a different approach than when encountering a crab. It may jet away using its siphon, releasing ink as a decoy cloud, or it may adopt a threatening posture, arms raised and body enlarged to appear more formidable. Ink itself is not merely a visual smokescreen. It contains chemicals that can interfere with a predator's sensory perception, confusing olfactory tracking. Released into water, it forms a pseudo-body shape that distracts attackers momentarily. That brief confusion is often sufficient for escape. Again, survival hinges not on overpowering the threat, but on manipulating its perception. Reproduction introduces a stark contrast. Many octopus species are semelparous, reproducing once before death. Females guard eggs for weeks or months without feeding, cleaning them, and ensuring oxygen flow with gentle currents generated by their arms. In such a period, cognitive capacity remains active even as the body deteriorates. The same intelligence used to solve environmental puzzles now maintains life in the next generation. When hatchlings emerge, they disperse into a world demanding immediate adaptability. Extreme conditions amplify the stakes of every decision. In shallow reefs, predators are numerous and competition intense. In the deep sea, scarcity and darkness dominate. In both realms, the octopus survives by exploiting what others cannot. Narrow spaces, temporary shelters, and momentary confusion. Strength is irrelevant when the body can reshape itself to avoid confrontation entirely. There is a deeper evolutionary twist embedded in this design. The absence of a rigid skeleton exposes the octopus to injury and vulnerability, yet it also grants regenerative capacity. Arms lost to predators can regrow over time. Neural plasticity allows behavioral adaptation across a lifetime. The very features that seem risky are offset by recovery mechanisms and learning ability. The ocean does not reward rigidity. It shifts, erodes, obscures. Pressure compresses, darkness conceals. In such an environment, predictability is fatal. The octopus answers with fluidity. Muscles substitute for bone, intelligence substitutes for armor, and flexibility substitutes for dominance. On a shadowed reef face, an octopus emerges cautiously from its den at dusk. The water is thick with plankton, light dim and fractured. It extends an arm into open water, testing currents, sampling chemical cues. A passing fish pauses, unaware of the subtle pattern shift that makes the octopus indistinguishable from stone. In a flash, arms extend and webbing unfurls. The prey is enveloped, subdued with venom delivered through the beak. Then the octopus retreats once more into darkness, sealing itself within its chosen crevice. What remains visible is not brute conquest, but quiet calculation. In extreme conditions where strength and speed are limited by pressure and scarcity, intelligence becomes the dominant survival trait. The octopus embodies a different kind of power, one rooted in adaptability rather than force. In the end, its survival strategy is not a single adaptation, but a network of them. Soft body, distributed brain, dynamic camouflage, chemical defense, regenerative tissue, behavioral flexibility. Each trait interlocks with the others, forming a system capable of responding to uncertainty. Where other animals resist the environment, the octopus conforms to it, reshaping itself physically and behaviorally as conditions demand. On the ocean floor, beneath layers of shadow and pressure, a shape dissolves into rock once more. There is no monument to its presence, no evidence of struggle, only a subtle shift in texture where muscle has become stone. In the harshest corners of the sea, survival is not about overpowering adversity. It is about becoming something the adversity cannot grasp. A fish lies in plain sight, and everything around it behaves as if it does not exist. Shrimp pick at the sand inches from its mouth. Small reef fish dart past its dorsal ridge. Even a cautious predator glides overhead without altering course. Then, in less time than a blink, the sand explodes and something vanishes between jaws that never seemed open. The most venomous fish in the ocean has just fed, and no one saw it move. The stonefish does not stalk. It does not chase. It becomes terrain. Pressed against coral rubble in shallow tropical waters, its body mimics erosion itself. Warty skin, mottled in browns and greys, echoes the textures of encrusted reef. Algae colonize its surface. Sediment settles along its flanks. It does not shake this covering off. It cultivates it. Over time, the line between animal and environment dissolves. These reefs are not tranquil gardens. They are high-traffic intersections of predator and prey, where currents shift temperature and salinity daily, and sunlight pulses in broken patterns through rippled water. Exposure is constant. Unlike species that retreat into caves or bury completely in sand, the stonefish often sits openly on substrate, relying not on concealment through distance, but on deception through identity. It does not hide behind coral. It is coral. The secret lies in stillness. Most fish ventilate gills continuously with visible opercular movement. The stonefish reduces motion to the bare minimum. Its gill openings are small and strategically placed, allowing respiration with minimal disturbance of surrounding water. Even its eyes, positioned high on the head, protrude subtly without betraying the outline of a predator. When it breathes, it does so quietly, without plume or pulse that would alert passing prey. Yet stillness alone would not suffice in such an exposed zone. The reef is full of sharp edges, abrasive sand, fluctuating oxygen levels, and sudden temperature changes during tidal cycles. The stonefish tolerates these shifts with physiological resilience. Its thick, scaleless skin forms a barrier against minor abrasions and parasite intrusion. Beneath that skin, muscle mass is compact and dense, enabling explosive action after prolonged inactivity. It can remain motionless for hours, conserving energy, then unleash a strike driven by rapid buccal expansion that creates a suction vortex. Water and prey are drawn inward before the victim has time to react. But the true insurance policy against the dangers of exposure stands along its dorsal ridge. 13 stout spines rise like jagged rocks along its back, each connected to venom glands at the base. These spines are not passive needles, they are pressure-activated delivery systems. When stepped on or bitten, the sheath surrounding each spine depresses, forcing venom upward through grooves and into the attacker's tissue. The toxin is a potent cocktail of proteins that disrupt cell membranes, interfere with neuromuscular transmission, and trigger intense pain and tissue damage. For a predator, the cost of a mistaken bite can be severe, even lethal. The reef contains many venomous species, yet the stonefish's strategy is unique in its reliance on invitation rather than warning. Brightly colored lionfish advertise their spines. Cone snails conceal theirs, but remain mobile. The stonefish blends completely, accepting the risk that something might contact it unknowingly. This paradox reveals a deeper strategy. By appearing as harmless substrate, it reduces encounters altogether. The venom becomes a final line of defense, not the primary deterrent. Predators learn quickly. A juvenile shark that samples a stonefish rarely repeats the experiment. Scar tissue and memory reinforce avoidance. Over evolutionary time, such interactions sculpt predator behavior around the stonefish's presence. In this way, venom does more than defend. It shapes the behavioral landscape of the reef. Certain microhabitats become de facto safe zones for the stonefish because experienced predators bypass them. Yet venom production is metabolically expensive. Synthesizing complex toxins demands energy and biochemical resources. The stonefish cannot afford constant regeneration of spent venom. This constraint reinforces its reliance on camouflage and immobility. Every unnecessary discharge is a cost. Efficiency governs its existence. Tides introduce another layer of stress. In shallow reef flats, water levels can drop dramatically during low tide, leaving pools isolated and temperatures rising under tropical sun. Oxygen concentrations fluctuate as photosynthesis slows or accelerates with light changes. Many fish retreat to deeper channels. The stonefish often remains. Its tolerance for low oxygen and variable salinity allows it to persist where others vacate. Reduced competition follows. Prey species seeking refuge in shrinking pools may find themselves sharing space with what appears to be an inert rock. The illusion extends beyond coloration. Skin appendages break up its outline, creating irregular contours that disrupt the perception of symmetry. To a fish scanning for predators, symmetry is a cue. The stonefish denies that cue. It adopts asymmetry, roughness, unpredictability in form. Evolution has refined this mimicry across species, each matching specific reef substrates within their geographic range. There is a subtle reversal embedded in this strategy. Most predators advertise vitality through movement. They display agility, speed, or pursuit. The stonefish survives by suppressing all signs of life. It trades dynamism for invisibility. Its success rate in ambush is high precisely because prey species filter out non-moving objects as irrelevant. In a reef crowded with shapes, stillness is misclassified as safety. Hunting mechanics reveal further precision. When a prey fish wanders within range, the stonefish does not lunge forward. Instead, it expands its buccal cavity at remarkable speed, generating negative pressure. Water rushes inward, carrying the prey with it. The jaws close instantly, and excess water exits through the gill openings. The entire event can occur in a fraction of a second. No chase, no struggle in open water, no extended expenditure of energy. After feeding, it resumes its role as terrain. Digestion proceeds slowly, matching its low metabolic baseline. It can endure extended periods without food, another adaptation suited to variable prey availability. Energy conservation underpins every aspect of its design. Environmental changes driven by warming seas and habitat degradation introduce uncertainty. Coral bleaching alters substrate coloration and texture, potentially affecting camouflage effectiveness. Sediment runoff from coastal development modifies reef composition. The stonefish's reliance on precise visual mimicry ties its fate to reef integrity. If the background changes faster than evolutionary adaptation can track, the illusion may falter. Yet the species has persisted across millennia of environmental fluctuation. Its broad tolerance for exposure and its dual strategy of invisibility and venom defense provide flexibility within limits. Juveniles may occupy slightly different habitats than adults, dispersing risk across environmental gradients. This plasticity suggests resilience, though not invulnerability. In the broader reef system, the stonefish plays a quiet regulatory role. By preying on small fishes and crustaceans, it influences local population dynamics. Its presence alters spatial behavior of prey species, encouraging vigilance and shaping micro-habitat use. Predators that avoid stonefish territories concentrate elsewhere, redistributing pressure across the reef. An animal that barely moves exerts influence disproportionate to its activity. As afternoon light shifts and shadows lengthen across coral rubble, the stonefish remains unchanged in posture. A thin layer of sand accumulates along its flank, further erasing its outline. A passing fish hesitates, then resumes feeding among algae. Somewhere nearby, a diver scanning the reef might mistake the predator for stone, until a subtle blink betrays its presence. The reef is a place of constant motion, yet survival here does not always belong to the swift. Sometimes it belongs to the patient, the venomous, and the indistinguishable. Motionless amid chaos, armored not with plates but with toxin, the stonefish endures heat, exposure, and scrutiny by refusing to appear alive at all. In a world where attention often means death, it thrives by becoming background. And as currents sweep over its ridged back and life flows around it unaware, the line between predator and landscape disappears once more. A lobster sheds its entire body and walks away from itself. The old shell splits open along the back, and from that fracture, something pale and soft pulls free, leaving behind a perfect hollow duplicate. For a brief and dangerous window, the animal that moments ago wore armor like a knight is reduced to a pliable, defenseless form hiding in darkness. This is not a rare event. It is the only way it survives. On the seafloor, longevity is not guaranteed by speed or dominance. It is negotiated through cycles of growth and exposure. The lobster does not stop growing after reaching maturity. Its tissues continue to expand year after year, and unlike many vertebrates, it does not exhibit the same predictable patterns of biological aging. Cells repair efficiently. Telomerase, an enzyme linked to cellular maintenance, remains active in many tissues throughout its life. In theory, a lobster could persist for decades, even beyond half a century, if nothing else intervenes. But something always intervenes. The Atlantic seafloor is a mosaic of rock outcrops, sand patches, kelp forests, and shadowed crevices. Within this terrain, the lobster navigates with sensory antennae extended, sampling chemical traces in the water. It is an opportunist, feeding on mollusks, fish carrion, worms, and sometimes smaller crustaceans. Its claws are asymmetrical, one optimized for crushing, the other for cutting. It is well defended, encased in a rigid exoskeleton composed of chitin reinforced with calcium carbonate. This armor protects against predators and environmental abrasion, yet that armor is also a prison. Because the lobster's skeleton is external, growth cannot occur gradually as in vertebrates. Instead, the entire shell must be discarded and replaced. The process begins long before the visible split. Beneath the old exoskeleton, a new soft shell forms. Calcium is withdrawn from the existing armor and stored internally in gastroliths, temporary mineral deposits. The lobster reduces feeding as the new shell thickens. Hormonal signals regulate the timing, aligning molt cycles with seasonal temperature shifts and energy availability. Then the rupture occurs. The lobster absorbs water rapidly, swelling its body to crack the old shell along predetermined weak lines. It flexes and pulls, wriggling backward until antennae, claws, and legs slide free. The act requires enormous energy. If any limb becomes stuck, the lobster may autotomize, voluntarily detaching the trapped appendage to escape. It can regrow that limb over subsequent molts, but the immediate cost is high. For hours, sometimes days, the lobster remains hidden. Its new exoskeleton is soft and vulnerable. Predators sense weakness in the water. Fish, octopus, and even other lobsters exploit molting individuals if discovered. The seafloor during molting season becomes a landscape of silent ambushes. This is the central paradox of lobster longevity. To grow and remain biologically young, it must periodically become defenseless. As lobsters age and increase in size, molting becomes less frequent but more taxing. A young lobster may molt several times in a year, each time adding measurable growth. An older individual might molt only once every few years. The energy demands scale upward with size. Producing a larger exoskeleton requires more material, more stored minerals, more metabolic expenditure. Recovery takes longer. Here lies the hidden limit. Lobsters do not die of simple old age in the way mammals do. They die because the process of molting becomes too costly or too risky. A very large lobster must generate sufficient force to escape its shell. If muscular strength or energy reserves are inadequate, it may fail mid-molt, trapped within its own armor. In other cases, the stress of molting can trigger physiological collapse. Growth, which once extended life, eventually becomes a liability. There is also the burden of mass. Larger lobsters are more visible. They require more oxygen, more food, and more shelter space. Competition intensifies. In some regions, dominant individuals claim prime crevices and aggressively defend them. Social hierarchies emerge through ritualized claw displays and chemical signaling. Subordinates yield without full combat, conserving energy. But as a lobster grows, it must continually reassert dominance or seek new territory. Chemical communication underpins much of this interaction. Urine released from specialized openings near the antennae carries pheromones that signal status and reproductive condition. During encounters, lobsters engage in antennae fencing, sampling each other's chemical cues before escalating. These signals reduce unnecessary fights, but they also create a network of recognition within dense populations. Reproduction introduces another layer of strain. Females carry fertilized eggs beneath their tails for many months, fanning them to provide oxygen. During this brooding period, they may reduce feeding and movement, increasing vulnerability. Once larvae hatch, they enter a planktonic phase, drifting in open water before settling to the bottom. Mortality in early life stages is immense. Longevity in adulthood is balanced by fragility in youth. Regeneration appears to offer resilience. Lost claws regrow over successive molts, sometimes larger than the original if the injury occurred early in life. This regenerative capacity enhances survival after predatory encounters. But regeneration requires additional resources during molting, further increasing the energetic burden. Environmental conditions complicate the equation. Rising ocean temperatures accelerate metabolism, increasing energy demands. Acidifying waters interfere with calcium carbonate deposition, potentially weakening exoskeleton formation. Pollutants accumulate in benthic zones where lobsters forage. Each of these factors interacts with the molting cycle, altering its timing and success. The evolutionary strategy of indeterminate growth, continuing expansion without a fixed adult size, offers advantages. Larger lobsters are less vulnerable to many predators. Their claws become formidable, capable of cracking thicker shells and deterring rivals. Size correlates with reproductive output in females, increasing egg production. But the cost curve rises with each incremental gain. In colder waters, metabolism slows, extending intervals between molts and potentially lengthening lifespan. In warmer regions, faster growth may shorten it. Longevity is not a static trait. It is an outcome shaped by environmental pressure and resource availability. Beneath a rocky ledge, an old lobster rests, its shell scarred from previous molts. It has survived numerous cycles of vulnerability. Its antennae sway gently, detecting faint currents. Each breath draws water across gills hidden beneath the carapace. The shell it wears is both shield and constraint. The common assumption is that aging is inevitable decline. For the lobster, decline is less about cellular decay and more about mechanical and energetic thresholds. As long as it can successfully molt, it can continue growing and repairing. The moment that balance tips, survival falters. The broader ecological impact of lobster longevity is significant. Long-lived individuals stabilize benthic communities through sustained predation and competition. Their burrowing activity alters sediment structure. Their feeding patterns influence populations of mollusks and other invertebrates. Remove large lobsters from an ecosystem and trophic cascades ripple outward. Human harvest has historically targeted the largest individuals, removing those that have survived multiple molting cycles. This selection pressure alters population structure, skewing toward younger, smaller lobsters. The absence of elders reshapes reproductive dynamics and social interactions. On a moonlit seafloor, the faint outline of an empty shell drifts among seaweed. Nearby, its former occupant, newly hardened, resumes patrol. Its new armor gleams slightly before algae begin to colonize once more. The cycle resets. Longevity here is not immortality. It is endurance through repetition of risk. Growth offers strength but demands surrender. Regeneration restores limbs but consumes energy. Each molt is a gamble, each survival a renewal of debt. In the silent dark beneath shifting currents, a lobster continues forward, carrying the weight of its next molt within. It does not age in the way most creatures do. Instead, it negotiates survival through a series of calculated vulnerabilities, growing larger, harder, and more exposed with every cycle. In the end, the limit is not time. It is the cost of shedding the shell that keeps it alive. The blow lands with the force of a small-caliber bullet, and the water itself flashes with light. A mantis shrimp has just struck, and for a fraction of a second the sea around its target erupts in collapsing bubbles, hot enough to emit a faint spark. The shell of a crab fractures instantly, but the real mystery is not the broken armor. It is the attacker's limb. How does a creature no longer than a human hand repeatedly unleash impacts that would shatter bone without destroying its own body? On a tropical reef flat, beneath broken coral slabs and rubble fields, a mantis shrimp waits at the mouth of its burrow. Its eyes swivel independently, scanning the surroundings with trinocular vision in each eye. They detect polarized light, subtle color gradients, minute movements in shifting sand. But the eyes are not the weapon. Folded tightly beneath its thorax are specialized appendages known as raptorial clubs, compact structures coiled like loaded springs. When prey wanders within range, the mantis shrimp does not lunge forward. It unfolds its strike in a motion so rapid that it challenges the limits of biological material. The appendage rotates outward, accelerating through water with blistering speed. The club contacts the prey with such force that not only does the shell crack, but the shockwave creates cavitation bubbles. As these bubbles collapse, they generate secondary bursts of energy that add to the destructive effect. It is a 1-2 punch, driven by both muscle and fluid physics. The question becomes unavoidable. Repeated exposure to such impact should damage tissue irreparably. Muscle fibers should tear. Bone equivalents should fracture. But the mantis shrimp's club is neither bone nor simple chitin. It is a composite material engineered through evolution to manage extreme stress. At the core of the club lies a region known as the impact surface, composed of highly mineralized hydroxyapatite arranged in a layered structure. Beneath this lies a helicoidal arrangement of chitin fibers embedded in a protein matrix, often described as a boulegen structure. These fibers are stacked in rotating layers, each oriented slightly differently from the one above. When a crack begins to form under stress, it encounters layers that redirect and dissipate its energy. Instead of propagating straight through, the crack twists and spreads, losing momentum. This architecture mirrors advanced human-engineered materials designed to resist fracture, yet it evolved naturally under the pressure of survival. The mantis shrimp's club does not simply withstand impact. It channels it, distributing force across microstructures that prevent catastrophic failure. Yet even this structural brilliance would not suffice without precise control. The strike is powered by a latch and spring mechanism within the appendage. Muscles slowly contract, storing energy in elastic structures while a latch holds the limb in place. When released, The stored energy converts rapidly into kinetic motion. This separation of energy storage and release allows the shrimp to generate force beyond what muscle contraction alone could achieve. The extreme acceleration subjects the club to tremendous mechanical stress. But because energy release is brief and controlled, tissues avoid prolonged strain. The entire strike lasts only a fraction of a second, minimizing cumulative exposure. In physics terms, the mantis shrimp operates at the boundary between elastic resilience and material failure, but it rarely crosses that threshold. There is another hidden layer of protection. After impact, the club experiences vibrations and shock waves that travel back through the appendage. Specialized regions within the limb absorb these waves, preventing them from damaging sensitive joints and muscles. The design is not merely about delivering force outward, it is about safeguarding inward. On the reef, the mantis shrimp's burrow is more than shelter. It is a fortress carved into substrate, often reinforced with sand and shell fragments. Here, the shrimp retreats between hunts. It must balance aggression with caution. Its weapon makes it formidable, but it is not invincible. Larger predators, including fish and octopus, can overpower it if caught in open water. The burrow offers refuge and control over engagement distance. Interestingly, not all mantis shrimp are smashers. Some species are spearers, equipped with barbed appendages designed to impale soft-bodied prey. This divergence illustrates an evolutionary trade-off. Smashers invest in mechanical resilience and brute impact to break hard shells. Spearers optimize for precision and speed against evasive fish. The smashing lineage chose to confront physics directly. Each strike carries risk. If the club were to fracture, regeneration would be possible over molting cycles, but during that period the shrimp would be vulnerable. Therefore, the frequency and context of strikes are strategic. The mantis shrimp does not waste its weapon. It evaluates prey size, shell thickness, and angle before committing. There is a remarkable sensory integration behind this decision-making. The shrimp's eyes provide detailed information about prey position and orientation. Its nervous system processes this data rapidly, coordinating timing and force. The strike must connect precisely. A glancing blow wastes energy and increases stress on the club without ensuring a kill. Mechanical stress also shapes life history. During molting, when the exoskeleton softens and is shed, the raptorial appendages lose their hardened impact surfaces. The shrimp becomes temporarily incapable of delivering full-strength strikes. It retreats deeply into its burrow, sealing the entrance. Survival during this period depends on prior success and energy reserves. The weapon that defines its ecological role must be rebuilt from scratch. As ocean conditions shift, additional stresses emerge. Increasing acidity in seawater can affect the availability of calcium carbonate, a key component in exoskeleton formation. While the mantis shrimp's club relies heavily on chitin and hydroxyapatite, overall exoskeletal integrity remains linked to environmental chemistry. The physics it operates against are constant, but the material resources required to counter them may fluctuate. Beyond predation, the strike serves in territorial disputes. Rival mantis shrimp engage in ritualized displays, sometimes striking each other's clubs in controlled bouts. These contests test strength and resilience without immediate lethal outcome. The sound of impact echoes faintly underwater, a percussive assertion of dominance. Even here, the same structural adaptations protect both participants from self-destruction. There is a profound irony embedded in this biology. The mantis shrimp is small, yet it manipulates forces that rival mechanical devices designed by humans. It does not exceed the limits of physics. It exploits them. Cavitation bubbles collapse at temperatures briefly hotter than the surface of the sun, though on microscopic scales. The shrimp does not generate that heat directly. It leverages fluid dynamics, creating conditions where physics completes the task. Repeated exposure to such extremes demands precision engineering at the biological level. The club's outer layer is sacrificial, capable of absorbing micro-damage that can be repaired during molting. Beneath that, the helicoidal layers remain intact, preserving structural integrity. This multi-layered defense mirrors shock-absorbing systems in advanced composites, where surface wear is expected but core stability is maintained. The mantis shrimp survives not by avoiding mechanical stress, but by mastering it. Every strike pushes against the boundary where material failure could occur. Every successful blow reinforces the evolutionary design that makes the next one possible. On the reef floor, another crab approaches the burrow entrance, claws probing cautiously. Inside, the mantis shrimp waits, coiled. The appendage unfolds again in a blur. Sand erupts. The crab's shell splits along a fault line invisible seconds earlier. The shrimp drags its prey inside, leaving no sign of the violence except a fractured carapace. Physics governs this world. Water resists motion. Materials fracture under stress. Energy dissipates. Yet within these rules, the mantis shrimp has carved out a niche at the very edge of possibility. It survives where mechanical limits threaten to destroy it, protected by structures that bend, redirect, and absorb the consequences of its own power. In the dim light filtering through coral rubble, the mantis shrimp remains poised at its threshold. Its club rests folded beneath its body, quiet now, but charged with stored potential. It lives on the frontier where biology meets brute force, striking again and again without breaking itself. A silver torpedo tears through open ocean at highway speed and does not slow down. Minutes pass, then hours. The fish does not glide to rest, does not sink into stillness. It keeps moving, muscles burning, heart hammering, water splitting around a body built for relentless motion. In a realm where most fish drift and dart in short bursts, the tuna lives in permanent acceleration. The danger is not a predator. The danger is stopping. Far from reefs and coastal shelter, the open ocean is a blue desert shaped by distance and scarcity. There are no walls to hide behind, no rocks to wedge into. Prey forms shifting clouds, predators patrol vast territories, and currents carve invisible highways through water columns that plunge into darkness. Here, survival favors those who can cover enormous distances efficiently. The tuna has answered this demand with one of the most extreme endurance systems in the marine world. Its body is a study in streamlining. From the pointed snout to the crescent-shaped tail, every contour reduces drag. Fins fold into grooves along the body at speed, minimizing turbulence. The tail, rigid and scythe-like, oscillates with minimal wasted motion, transferring energy directly into forward thrust. But hydrodynamics alone do not explain how a tuna maintains velocity that would exhaust most fish in seconds. The deeper secret lies in heat. Unlike the majority of bony fish, tuna are regionally endothermic. They can elevate the temperature of their swimming muscles above the surrounding water. This is not full warm-bloodedness in the mammalian sense, but it is a remarkable deviation from typical ectothermic design. Warm muscle contracts more powerfully and more rapidly. Enzymatic reactions involved in metabolism operate more efficiently at higher temperatures. In cold waters where other fish grow sluggish, tuna maintain performance. This internal warmth is generated by continuous muscle activity and preserved through a countercurrent heat exchange system known as the rete mirabile. Blood vessels leaving the red swimming muscles run alongside vessels returning from the gills. Warm blood flowing outward transfers heat to cooler blood returning inward. The result is conservation of metabolic heat within the core musculature. Instead of losing warmth immediately to the surrounding sea, the tuna traps it, creating a thermal pocket inside its body. The effect is transformative. Elevated muscle temperature enhances aerobic capacity. Red muscle fibers, rich in mitochondria and myoglobin, dominate the tuna's body composition. These fibers are specialized for sustained contraction, fueled by oxygen-dependent metabolism. Unlike white muscle fibers used for short bursts, red fibers support endurance. But warm muscle demands fuel and oxygen in immense quantities. The tuna's circulatory and respiratory systems are scaled accordingly. Its heart is proportionally large, pumping blood at high pressure to deliver oxygen rapidly. Gills are expansive, with a high surface area for gas exchange. As water passes over delicate gill filaments, oxygen diffuses into blood efficiently. replenishing what is consumed by constantly active tissues. In many fish, bursts of speed rely partly on anaerobic metabolism, producing lactic acid and requiring recovery periods. Tuna operate primarily aerobically, even at high speeds. This means they avoid the rapid fatigue associated with anaerobic bursts. They can cruise swiftly without accumulating metabolic debt. Yet this system has a strict requirement. Water must keep moving across the gills. Tuna are obligate ram ventilators. They cannot effectively pump water over their gills while stationary. If they stop swimming, oxygen intake drops precipitously. Movement is not optional, it is life support. In this sense, the tuna is both master and prisoner of speed. Across the pelagic zone, schools of tuna move like coordinated missiles. Grouping provides hydrodynamic and strategic advantages. Fish in the center of a school experience slightly reduced drag, conserving energy. At the same time, collective movement confuses prey and deters predators. But schooling also requires constant adjustment. Each individual must maintain position relative to neighbors, matching speed and direction without collision. Neural processing and sensory feedback operate continuously. Prey often consists of smaller fish and squid that themselves rely on speed and agility. When tuna hunt, they accelerate further, herding prey into dense balls near the surface. In these feeding frenzies, speed becomes weaponized. The school corrals its target, reducing escape routes. Dolphins and seabirds may join the assault, capitalizing on the chaos. The hunt illustrates a paradox. Tuna are built for endurance, yet they can unleash bursts approaching their physiological ceiling. The same warm muscle that sustains cruising allows rapid acceleration. But pushing too close to maximum output increases energy expenditure dramatically. The margin between sustainable speed and exhaustion narrows. Strategic pacing becomes critical. Migration reveals the scale of this endurance. Some species traverse entire ocean basins, following temperature gradients and prey availability. These journeys span thousands of kilometers. Warm-blooded muscle enables them to exploit colder waters where competitors falter. It expands their ecological range. But energy remains the governing currency. Maintaining elevated muscle temperature requires constant fuel intake. Tuna feed voraciously to support their metabolism. A decline in prey abundance can have cascading effects. Without sufficient calories, endurance falters. Growth slows. Reproductive output declines. Reproduction itself demands additional reserves. Many tuna spawn in warm waters, releasing vast numbers of eggs into currents. Larvae face extreme mortality, relying on sheer numbers for species persistence. Adults must balance the energetic demands of migration, hunting, and gamete production. There is a limit imposed by physics and physiology. Drag increases exponentially with speed. Doubling velocity requires far more than double the energy. The tuna's streamlined form mitigates this but cannot eliminate it. Efficient propulsion and heat retention allow sustained cruising, yet maximal speed remains costly. The ocean environment adds complexity. Temperature stratification affects muscle performance. In colder depths, metabolic rates drop unless heat is conserved effectively. Oxygen levels vary with depth and region. Hypoxic zones challenge endurance specialists by limiting oxygen availability. Tuna often adjust depth to balance temperature and oxygen constraints. Predators such as sharks and billfish pursue tuna, but the tuna's sustained speed offers a significant defensive advantage. Instead of relying solely on maneuverability, it escapes by maintaining velocity beyond the comfortable range of many pursuers. Endurance becomes armor. Fishing pressure introduces a modern twist. Industrial fleets target large schools, exploiting the tuna's predictable migratory routes. The very traits that make tuna successful in open ocean ecosystems make them vulnerable to coordinated human capture methods. Long lines and purse seines intercept moving schools efficiently. The removal of large, mature individuals alters population dynamics and migratory patterns. Still, in the vastness beyond fishing fleets, the tuna's design remains a triumph of evolutionary engineering. Warm muscle sustained by countercurrent heat exchange, red fiber dominance, high-capacity gills, and a powerful heart create a biological engine tuned for constant motion. As twilight deepens over open water, A school of tuna glides just beneath the surface. Their bodies shimmer silver and blue in fading light. They do not pause. Even in apparent calm, their tails sweep rhythmically, propelling them forward. Beneath them lies abyssal darkness. Above, the sky mirrors the sea. The open ocean offers no refuge for stillness. To survive here is to move, to convert oxygen and fuel into momentum without collapse. The tuna has embraced that demand completely, operating at the boundary where energy supply and expenditure remain in perfect balance. In the endless blue, they continue their forward surge, warm hearts beating in cold water, living proof that endurance, not brute force, can dominate the horizon. A Moray eel appears to be snarling, jaws gaping open in a silent threat, teeth exposed in permanent aggression. But the truth is more urgent than intimidation. It is not baring its fangs to warn a rival. It is fighting to breathe. Deep within a coral reef, in a crevice so narrow that sunlight filters in only as fractured shards, oxygen is scarce. Water here moves slowly, trapped between stone and sponge, warmed by tropical heat and depleted by respiration of countless hidden organisms. Fish that venture too long into these cavities risk suffocation. Yet this is where the Moray eel chooses to live. The reef above pulses with movement. Parrotfish grind coral. Surgeonfish graze algae in shimmering schools. Currents sweep across open faces, bringing fresh, oxygenated water. But beneath the surface layers, inside the reef's skeleton, space compresses and ventilation falters. This labyrinth of cracks and tunnels is both refuge and hazard. The Moray eel thrives precisely where the water struggles to circulate. Its body is elongated and serpentine, built for penetration rather than speed. It slips through coral corridors that would trap broader fish, curling into dens that shield it from larger predators. But unlike many reef dwellers, the Moray lacks an operculum, the bony gill cover that most fish use to pump water across their gills while stationary. Instead, it must rely on opening and closing its mouth to force water over delicate respiratory surfaces. This constant gaping, often misinterpreted as aggression, is a survival necessity. By rhythmically expanding and contracting its buccal cavity, the Moray draws water in and pushes it across its gill slits, maintaining oxygen exchange even in stagnant pockets. The motion is deliberate, almost mechanical. Open, close, open again. Without this active pumping, oxygen levels within its bloodstream would drop rapidly. The environment demands this specialization. Coral reefs are oxygen-rich at the surface during daylight, when photosynthetic algae release oxygen. But at night, or deep within crevices where light barely penetrates, respiration by corals, sponges, and microorganisms can reduce oxygen concentration significantly. In these microhabitats, survival depends on extracting every possible molecule of dissolved oxygen from slow-moving water. The Moray eel's gill surface area is proportionally efficient, optimized for high extraction rates. Blood flow through its gill lamellae maximizes diffusion gradients. But even efficiency has limits. If oxygen levels fall too low, the eel must adjust behavior, sometimes extending its head further into open water while keeping most of its body concealed. This partial exposure balances safety with respiratory necessity. Its lifestyle is a trade-off between protection and ventilation. Tight crevices shield it from sharks and large predatory fish. Yet those same walls restrict water flow. The Moray solves this dilemma by positioning itself strategically, often aligning its body so that even minor currents enter its den. In reef zones where tidal flows pulse predictably, the eel selects locations that experience periodic flushing. Hunting in such confined spaces introduces another layer of complexity. The Moray does not rely solely on vision. Within the shadows of its lair, it depends heavily on olfaction. Chemical cues carried by water reveal the presence of injured fish or hidden crustaceans. Its nostrils, extended as tubular projections, sample these signals continuously. Even in low visibility and low oxygen, scent becomes a map. When prey approaches, the Moray's strike is swift and precise. Its jaws close with force, but the true marvel lies deeper. Moray eels possess a second set of jaws, pharyngeal jaws, capable of lunging forward within the throat to grasp prey and pull it inward. This adaptation compensates for the eel's elongated body, which lacks the suction-feeding capacity of many reef fish. In tight spaces where maneuverability is limited, the pharyngeal jaws secure meals efficiently without requiring the eel to retreat or reposition. This feeding strategy is tightly linked to its oxygen management. A prolonged struggle could compromise respiration, especially in poorly ventilated dens. By securing prey quickly and reducing thrashing, the eel conserves oxygen and avoids drawing attention from competitors. But the reef is not static. Oxygen levels fluctuate with temperature and time of day. During heat waves, warmer water holds less dissolved oxygen. Coral bleaching events alter community structure, affecting nighttime oxygen consumption. The Moray's resilience is tested by these environmental changes. Its dependence on active ventilation means any prolonged drop in oxygen availability can be critical. Interestingly, some morays engage in cooperative hunting with groupers, a rare interspecies collaboration. In open reef zones, groupers signal to morays through body language, indicating prey hidden within crevices. The moray flushes prey from tight spaces, driving them into open water where the grouper can strike. In this partnership, oxygen constraints influence tactics. The moray cannot chase prey across open expanses indefinitely. Its strength lies in penetrating confined spaces where ventilation remains manageable. Despite their fearsome appearance, moray eels are not reckless. Energy conservation governs much of their behavior. They spend extended periods resting within dens, reducing metabolic demand. Their elongated musculature allows efficient undulatory swimming when necessary, but they avoid unnecessary excursions. Each movement must justify the oxygen it consumes. There is an irony in their intimidating display. The open mouth and visible teeth signal danger to potential threats, discouraging intrusion. Yet that same posture is a sign of vulnerability. The moray must expose itself slightly to breathe. What appears as menace is often a biological compromise. Predators that target morays exploit this constraint. In rare instances, larger fish or sharks may attempt to extract an eel from its den. The eel's narrow refuge offers protection, but if forced into open water, it loses its respiratory advantage. Rapid swimming demands higher oxygen intake, pushing its ventilation system to the edge. The internal physiology reflects adaptation to these demands. Hemoglobin in moray blood binds oxygen efficiently, even at lower concentrations, extending tolerance to mildly hypoxic conditions. However, extreme oxygen depletion still poses lethal risk. The eel's survival strategy depends on maintaining microhabitats where oxygen flow remains just sufficient. As dusk settles over the reef, oxygen dynamics shift. Photosynthesis ceases. Respiration dominates. Levels drop subtly within crevices. The moray's mouth opens wider, pumping steadily. Its gills work harder to extract diminishing reserves. The reef quiets, but inside its hidden corridors, a silent battle unfolds between metabolism and environment. The larger ecological implication is profound. By occupying low oxygen refuges, moray eels reduce competition with more oxygen-dependent predators. They exploit niches that others avoid. This specialization shapes prey distribution and predator-prey interactions within reef systems. In a narrow coral tunnel, the moray remains coiled, only its head visible. Its jaws part rhythmically, water flowing in and out in measured pulses. Around it, the reef breathes as well, currents ebbing and returning, oxygen rising and falling with cycles of light and tide. The limit here is invisible, yet absolute. Without continuous flow across its gills, the moray cannot survive. Its life is defined by the movement of water through its mouth. Every breath is an act of engineering within a confined world. In the dim recesses of the reef, where stone meets shadow and water barely stirs, the moray endures. It does not conquer oxygen scarcity. It negotiates it, mouth open, teeth bared not in threat, but in necessity, surviving on the narrow margin where water still carries enough life to sustain another breath. A translucent cube drifts just beneath the surface, nearly invisible, its body no thicker than glass. It has no brain, no centralized command center, no skull to protect thought. And yet it is about to execute one of the most precise hunts in the ocean. In shallow, tropical waters, where mangrove roots tangle like skeletal fingers and sunlight fractures into shimmering columns, a box jellyfish hovers with subtle control. It does not drift aimlessly like its distant cousins. Its movements are deliberate. It adjusts orientation. It avoids obstacles. It maintains position against mild current. For an animal built from gelatin and nerve nets, this level of coordination appears improbable. Most jellyfish operate as passive drifters, at the mercy of currents, responding reflexively to touch or light. The box jellyfish, however, belongs to a different lineage within the cnidarians. Its bell is shaped like a cube, each corner trailing long, contractile tentacles armed with thousands of specialized stinging cells. But what sets it apart is not simply venom, it is perception. Embedded along the rim of its bell are sensory structures called rhopalia. Each rhopalium houses multiple eyes. Some detect only light intensity, others form crude images. Within these tiny sensory clusters lie lenses, retinas, and photoreceptive cells capable of distinguishing contrast and orientation. The jellyfish cannot process information in a centralized brain because none exists. Instead, its nervous system is distributed, a decentralized network where processing occurs locally. As the animal pulses forward, rhythmic contractions of its bell generate thrust. Unlike typical jellyfish that drift, box jellyfish actively swim, capable of surprising bursts of speed. They navigate toward darker silhouettes that signal mangrove roots, where small fish seek shelter. They avoid collisions by steering around obstacles. All of this emerges from a body plan lacking centralized cognition. The secret lies in distributed control. Each rhopalium connects to nerve rings encircling the bell. Sensory input from eyes feeds directly into local circuits that trigger motor responses. It is a biological system where perception and action are tightly coupled. There is no deliberation in the human sense, no abstract thought. Instead, there is rapid sensory integration at the edge of the body. Within this simplicity lies remarkable efficiency. A fish darts between roots, its scales reflecting fragmented light. The jellyfish adjusts its course slightly, tentacles trailing behind like invisible threads. As it approaches the mangrove cluster, it slows, maintaining distance from hard surfaces that could damage its delicate tissues. This precision is not accidental. Laboratory observations have shown that box jellyfish orient themselves relative to vertical structures, maintaining specific distances. Venom becomes relevant only at the final moment. The tentacles contact prey, and specialized cells called nematocysts discharge with microscopic force. Each nematocyst is a coiled harpoon loaded under osmotic pressure. Upon mechanical or chemical stimulation, the harpoon everts explosively, injecting venom into tissue. This process requires no brain. It is a cellular reflex embedded in the architecture of the tentacle. The venom itself is complex, containing toxins that target cardiac and neural systems. In small fish, the effect is rapid paralysis. In humans, accidental contact can cause extreme pain and potentially lethal cardiovascular effects. But for the jellyfish, venom is not aggression. It is an extension of simplicity. Rather than pursuing prey with jaws or teeth, it allows chemistry to do the work. There is an apparent contradiction in this design. A creature without centralized intelligence navigates a visually complex habitat, times its swimming patterns, and selects productive hunting grounds. The resolution lies in evolutionary tuning. Over millions of years, natural selection refined the coupling between sensory input and motor output. The jellyfish does not understand its environment. It embodies algorithms shaped by survival. During daylight, box jellyfish often retreat to deeper waters, avoiding exposure to predators and harsh light. At dusk, they migrate toward shallows, where fish larvae and small crustaceans gather. This daily movement pattern emerges from light-sensitive eyes that detect changes in intensity. The shift in position appears purposeful, yet it is triggered by simple thresholds encoded in neural circuits. What seems like limitation becomes advantage. A distributed nervous system is less vulnerable to catastrophic failure. Damage to one region does not incapacitate the entire organism. Reflex arcs remain intact. Function persists. The body is resilient precisely because it lacks centralization. Still, there are boundaries. Without a brain, the jellyfish cannot learn in the way vertebrates do. Its responses are largely pre-programmed. Complex social interaction, strategic planning, and memory are beyond its reach. Yet within its ecological niche, these higher functions are unnecessary. Its environment demands rapid detection of light gradients, obstacle avoidance, and effective prey capture. These tasks it performs with unsettling competence. A school of juvenile fish hovers near the mangrove edge. The jellyfish pulses forward, adjusting angle relative to vertical roots. It avoids direct impact, threading through open gaps. Tentacles extend into the fish cluster. A flash of movement, then stillness. The prey ceases struggling almost instantly. The jellyfish draws the immobilized body toward its mouth, located at the center of its bell. Feeding is simple but efficient. Enzymatic digestion begins externally as prey is transferred to oral arms. Nutrients are absorbed directly into the gastrovascular cavity. There is no circulatory system as in vertebrates. Diffusion distributes nutrients through gelatinous tissues. Oxygen uptake occurs across body surfaces and through thin tissues lining internal cavities. Energy efficiency defines its survival. Swimming requires rhythmic contraction of muscle cells embedded in the bell. Each pulse expels water, generating forward thrust. Between pulses, elastic recoil restores shape. This method conserves energy compared to constant muscular exertion. The simplicity of its body plan reduces metabolic demands, yet danger remains. Sea turtles, immune to many jellyfish venoms, prey upon them. Some fish species nibble tentacles with caution. Environmental fluctuations such as salinity shifts and temperature changes influence survival rates. In warming seas, box jellyfish populations may expand into new territories, altering local ecosystems. There is a subtle ecological implication in their success. As effective predators of small fish and larvae, box jellyfish can influence recruitment rates in coastal habitats. Their presence affects population dynamics of reef fish. Despite lacking complex cognition, they participate in intricate ecological webs. The deeper twist is that intelligence, as commonly defined, is not required for ecological dominance in certain contexts. The box jellyfish demonstrates that functional sophistication can arise from minimal neural architecture. Its decentralized nervous system is optimized for a narrow but demanding set of tasks. In a habitat of shifting light and dense structure, it navigates with efficiency. As night deepens, the mangrove shallows become a theater of subtle motion. The jellyfish continues its patrol, eyes detecting faint contrasts, nerve rings coordinating contraction. There is no thought behind the movement, no conscious intent. There is only integration of signal and response. In the end, biological simplicity does not equate to vulnerability. It represents a different solution. In the warm coastal waters, where complexity overwhelms many species, the box jellyfish survives through elegant reduction. No brain, yet perception. No strategy, yet success. A translucent hunter moving with silent precision, proving that evolution can build lethal competence from the barest neural blueprint. A sea urchin can survive being eaten alive, not metaphorically, not symbolically, physically. A predatory fish bites down, cracks off a cluster of spines, tears away soft tissue from the exposed surface, and swims off with a mouthful of living flesh. The urchin remains, damaged, open, bleeding coelomic fluid into the water, but still alive. In most animals, that kind of wound is fatal. In a sea urchin, it is Tuesday. Across rocky coastlines and coral reefs, sea urchins exist under permanent siege. Triggerfish patrol with crushing jaws built for armored prey. Sea otters dive with dexterous paws, cracking shells against stone anvils. Lobsters probe crevices with jointed claws. Even certain wrasses and pufferfish specialize in picking at exposed tube feet and chewing through the test, the rigid calcium carbonate shell that encases the urchin's body. There is no safe hour, no true refuge. The urchin does not flee, it endures. At first glance, the defense seems obvious. A sphere bristling with spines, sharp and mobile, radiating outward in every direction. But this is not a static barricade. Each spine is anchored by a ball and socket joint embedded in the shell, controlled by muscle fibers and connective tissue. The urchin can angle its spines toward contact, orienting a living hedge of deterrence around its body. When touched, neighboring spines converge on the point of pressure, increasing density where the threat is greatest. Yet spines alone are not the whole system. Between them lie thousands of tube feet, delicate hydraulic extensions connected to an internal water vascular system. These tube feet allow movement, attachment to rock, even manipulation of small objects. They also serve as sensory organs, detecting chemical cues of nearby predators. The urchin does not see or hear in a conventional sense, but it senses vibration and dissolved molecules in the water. A hunting triggerfish leaves a signature in the current. The urchin responds by tightening its posture, lifting spines, bracing against substrate. Still, predators adapt. Some fish flip urchins onto their oral side, where spines are shorter and the mouth is exposed. Otters exploit leverage and tools. Certain sea stars envelop urchins slowly, immune to spine penetration, digesting tissue externally. The reef and shoreline are classrooms of evolutionary pressure, and the urchin is a permanent student. The true core of its survival strategy is not avoidance. It is resilience. The shell, called a test, is constructed from interlocking plates of calcium carbonate arranged in geometric precision. These plates are not fused into a single slab but sutured together, allowing a degree of flexibility and distributing force across the structure. When a predator bites, cracks do not propagate easily. Damage localizes. Structural integrity remains. Beneath the shell lies mutable connective tissue, a remarkable biological material capable of rapidly altering stiffness. When threatened, the urchin can increase the rigidity of certain tissues, locking its body into a defensive configuration. When relaxed, those same tissues soften, allowing movement and growth. This capacity to tune mechanical properties in real time is rare among animals and crucial under constant attack. But even with spines, shell, and connective tissue, injuries occur frequently. Entire sections of spines can be lost in a single encounter. Portions of the test can fracture. Tube feet can be severed. In such an environment, survival depends on regeneration. Sea urchins possess extraordinary regenerative capacity. Spine bases contain specialized cells capable of rapid proliferation. When a spine breaks, cellular activity accelerates at the stump. Within days, a new spine begins to extend, restoring defensive coverage. Tube feet regrow. Even sections of damaged shell can be repaired through deposition of new calcium carbonate. This is not casual healing. It is metabolic investment. Regeneration consumes energy. Resources diverted to rebuilding cannot be used for growth or reproduction. Every defensive recovery carries cost. Yet in ecosystems where predation pressure is relentless, the ability to absorb damage and restore structure becomes more advantageous than escape speed or concealment. There is an ecological paradox embedded in this endurance. In many reef systems, sea urchins act as grazers, scraping algae from rock surfaces with a complex jaw apparatus known as Aristotle's lantern. This five-toothed structure grinds calcium carbonate substrate, maintaining open space for coral settlement. When predator populations decline, urchin numbers can explode. With fewer threats, their grazing intensifies, sometimes stripping reefs to bare rock. But when predators are abundant, urchin densities remain balanced. The constant attrition keeps grazing in check. The urchin's defensive endurance allows it to persist long enough to fulfill its ecological role, but not dominate unchecked. It is both victim and regulator. A surprising twist emerges when examining spine function more closely. Some urchins possess venomous spines, capable of injecting toxins that deter predators chemically, not just mechanically. Others carry pedicellariae, tiny pincer-like structures between spines that snap shut on contact. These micro-weapons can grasp, inject venom, or remove settling organisms from the shell's surface. The defensive perimeter is layered, extending beyond simple sharpness. And yet, despite this arsenal, many predators have evolved counters. Triggerfish flip and bite strategically between spines. Sea otters target the oral side. Certain crabs sever spines methodically before attacking the shell. The relationship is not static defense, but escalating arms race. The most remarkable element of urchin survival is temporal. They are built not for winning a single confrontation, but for surviving many. Their lifespan can extend decades in some species. Over that time, countless minor injuries accumulate and heal. Each regenerated spine is a record of past conflict. Energy budgeting becomes critical. Urchins feed primarily on algae and detritus, converting low-quality plant material into structural calcium carbonate and regenerative tissue. In nutrient-rich environments, regeneration proceeds efficiently. In degraded habitats, recovery slows. The limit of their defense is endurance, but endurance is constrained by energy availability. Climate change introduces new variables. Ocean acidification reduces the availability of carbonate ions needed to build and repair shells. Lower pH can weaken test integrity, making fractures more likely and regeneration more costly. In such conditions, the balance between predator pressure and defensive repair may shift dramatically. There is another hidden strength within their simplicity. Unlike animals reliant on complex behaviors for defense, sea urchins operate largely through mechanical and physiological processes. They do not need to learn predator tactics. They do not need to coordinate socially. Their survival strategy is embedded in tissue properties and regenerative programs. As long as energy flows and carbonate remains available, the system functions. On a wave-battered shoreline, a sea otter dives again, selecting another urchin from a cluster. The sound of shell cracking echoes underwater. Spines scatter. Tissue tears. The otter feeds and leaves fragments behind. Among the shattered remains, a partially consumed urchin clings to rock, its tube feet gripping stubbornly. Days later, new spines begin to rise from broken bases. It will not chase predators away. It will not outwit them. It will endure them. Across reefs and rocky coasts, millions of these armored spheres persist under constant assault. Their strategy is not invincibility, but recoverability. In a world where attack is inevitable, survival favors those who can be broken and rebuilt. Under the shifting light of shallow water, a sea urchin remains anchored to stone, its spines extended in silent defiance. Each one a temporary structure, each one replaceable. Defense here is not about avoiding harm. It is about surviving it, again and again, until the predators move on and the next generation rises from the rubble. The first touch should kill it. A juvenile clownfish drifts too close to a forest of swaying tentacles, each one armed with microscopic harpoons primed to fire. A single brush from those threads can paralyze a small fish in an instant. The tentacles close around prey, venom floods muscle tissue, and digestion begins before the victim fully understands what happened. Yet this fish does not recoil. It presses closer. It lets the tentacles graze its skin, and nothing happens. The sea anemone continues its patient ambush, tentacles extended into the current like a living minefield. Nearby damselfish avoid it. Butterfly fish hover at a cautious distance. Even reef predators give it space. The anemone's stinging cells, called nematocysts, are mechanical weapons triggered by contact and chemical recognition. They fire with explosive speed, injecting toxins that disrupt nerve and muscle function. For most reef dwellers, the anemone is untouchable. But within that lethal crown of tentacles, orange and white bands flicker in and out of view. A clownfish weaves through the forest, brushing against venomous filaments that would incapacitate almost any other fish on the reef. It darts, pivots, hovers, and then retreats deeper into the tentacles as a larger predator passes overhead. This is not recklessness. It is a chemical truce. The skin of a clownfish is coated in a specialized mucus layer. This mucus is not merely a lubricant or pathogen barrier. It is a biochemical shield. Sea anemones rely on molecular recognition to distinguish prey from self. Their nematocysts are triggered not only by mechanical contact, but by chemical cues on the surface of potential victims. The clownfish's mucus masks, or mimics those cues. When a juvenile clownfish first approaches an anemone, it does not rush in. It performs a delicate acclimation ritual, making tentative contact, retreating, touching again. Over time, it acquires elements of the anemone's own mucus, or suppresses triggering signals within its own. The result is a chemical disguise. The anemone no longer identifies the fish as prey. This relationship is often described as mutualism, but its foundation is biochemical compatibility. The clownfish gains refuge from predators. The anemone gains protection and nutrients. The fish chases away butterfly fish that nip at tentacles. It removes debris. Its waste products provide nitrogen compounds that fertilize symbiotic algae living within the anemone's tissues, enhancing photosynthetic output. The reef environment surrounding this alliance is a battlefield of predation. Small fish rarely enjoy secure shelter. Coral branches provide hiding spots, but not immunity. Rock crevices offer cover, but larger predators can extract occupants. The anemone, by contrast, is a fortress armed with venom. The clownfish has chosen to live inside the weapon. The paradox deepens when examining the toxins involved. Anemone venom contains a cocktail of peptides and proteins, targeting sodium channels in nerve cells, causing rapid depolarization and paralysis. In laboratory settings, exposure can incapacitate fish within seconds. The clownfish does not possess immunity in the sense of resistance to toxin once injected. Its survival depends on preventing nematocyst discharge altogether. The mucus layer acts as both camouflage and buffer. It is thicker than that of many reef fish and contains glycoproteins that alter surface chemistry. Some studies suggest that clownfish mucus lacks certain triggering compounds that stimulate nematocyst firing. Others propose that through repeated contact, the fish incorporates anemone-derived chemicals into its mucus, achieving molecular mimicry. This chemical tolerance has limits. Remove a clownfish from its host anemone and place it with another species of anemone, and the process must begin again. Not all clownfish can coexist with all anemone species. Compatibility is species-specific, shaped by coevolution. Over millions of years, certain pairs have fine-tuned their chemical languages to avoid lethal misunderstanding. Within the tentacles, social dynamics unfold. Clownfish live in strict hierarchies. A single dominant female occupies the largest position, accompanied by a breeding male and several smaller non-breeding individuals. The hierarchy is enforced through subtle aggression and size-based dominance. If the female dies, the breeding male undergoes a remarkable transformation. It changes sex, becoming female, while the largest subordinate matures into the new breeding male. This reproductive flexibility ensures continuity within the protective confines of the anemone. The shelter is limited in space and resources. Only one breeding pair can be supported effectively. The rest wait, constrained by growth suppression and social control. Survival here is not only about resisting venom, but navigating hierarchy. Predators circle beyond the tentacles. Groupers, wrasses, and snappers patrol the reef. A clownfish that strays too far risks immediate pursuit. When threatened, it retreats into the anemone, pressing its body against stinging tentacles that part harmlessly around it. The predator hesitates. Even species immune to mild stings avoid direct entanglement. There is a strategic dance in feeding behavior as well. Clownfish venture outward to collect plankton and small invertebrates carried by current. They remain tethered to the anemone's radius, rarely exceeding a short distance. This restricted foraging range limits resource access, but maximizes survival probability. It is a trade-off between nutritional abundance and predation risk. An unexpected twist lies in how the anemone benefits. Movement of the clownfish increases water circulation around tentacles, enhancing gas exchange. The fish's bright coloration may attract larger fish toward the anemone, increasing opportunities for prey capture. What appears to be a simple shelter arrangement becomes an integrated ecological unit. Yet this alliance is fragile. Rising ocean temperatures induce bleaching in anemones, disrupting the symbiotic algae that provide much of their energy. A bleached anemone becomes weakened, its tentacles less robust, its growth stunted. Clownfish dependent on that host may face increased exposure and reduced reproductive success. Chemical tolerance cannot compensate for habitat collapse. There is also evidence that increased carbon dioxide levels in seawater can alter fish sensory perception. In more acidic conditions, juvenile clownfish may struggle to detect predator cues effectively, increasing mortality during dispersal stages before they settle into anemones. The early life phase, drifting as plankton, remains perilous. The clownfish's survival strategy is therefore layered. It begins with biochemical adaptation, extends through social organization, and integrates into broader reef dynamics. Its bright coloration, often perceived as a warning or advertisement, contrasts sharply with the lethal neutrality of its host. Orange and white bands flicker among toxic tentacles, signaling occupancy to other clownfish, but offering no protection to outsiders. The deeper revelation is that toxicity here is not conquered but negotiated. The clownfish does not neutralize venom. It avoids triggering it. Its entire existence depends on maintaining chemical compatibility. A disruption in mucus composition, a stress-induced shift in physiology, or a change in host species can render it vulnerable. Within the swaying tentacles, a clownfish hovers as dusk settles over the reef. Predators retreat to deeper water. The anemone contracts slightly, tentacles folding inward. The fish presses closer, nestled against living threads of toxin. In a world where danger radiates from every direction, survival sometimes means inhabiting the threat itself. The clownfish has chosen a home that would kill almost anything else. Its protection lies not in armor or speed, but in chemistry. A thin layer of mucus separates life from paralysis, coexistence from catastrophe. As currents shift and plankton drift through the water column, the anemone remains anchored, venom ready. Within its grasp, the clownfish moves freely, a bright pulse of motion inside a toxic crown. Here, endurance is measured not in muscle or might, but in molecular precision. The limit is chemical tolerance, and at its edge, the clownfish thrives. The silver flash lasts less than a heartbeat. One moment the reef edge is calm, a loose cloud of sardines hovering in loose formation above coral heads. The next, the water detonates. Scales erupt in a glittering shockwave, bodies scatter in all directions, and something long, metallic, and impossibly fast slices straight through the center of the swarm. By the time the fish understand what happened, one of them is already gone. The barracuda does not stalk in the shadows like a reef ambush predator. It does not rely on camouflage or patient infiltration. It survives on a single violent advantage. Explosive acceleration so extreme that reaction time itself becomes irrelevant. In the open margins between reef and blue water, where prey can see danger coming from every direction, survival depends not on surprise but on speed beyond anticipation. A mature barracuda hangs almost motionless in the water column, body elongated and rigid, pectoral fins making subtle corrections. Its coloration is deceptive, a mirror of the surrounding sea. Dark dorsal shading fades into a pale underside, creating countershading that erases its outline from below and above. Its flanks reflect fragmented light, blending into the shimmer of open water. From a distance, it appears like a streak of current. But beneath that sleek exterior lies machinery built for violent release. The barracuda's body is a hydrodynamic missile. Its musculature is arranged for sudden contraction rather than prolonged endurance. Unlike tuna, which specialize in sustained high-speed cruising, barracuda rely on fast-twitch muscle fibers concentrated along the flanks. These fibers generate rapid bursts of force, but fatigue quickly. This predator does not chase for long distances. It annihilates space in an instant. A school of reef fish drifts along the drop-off, feeding casually in dappled sunlight. Each individual maintains a small buffer zone from its neighbors, relying on collective vigilance. The school functions as a sensory network. Hundreds of eyes scan for movement. Tiny lateral lines detect subtle vibrations. But the barracuda does not announce itself. When it strikes, it does not begin from zero. It primes. Its body arches slightly. Caudal fin stiffens. Neural signals cascade through motor neurons with brutal efficiency. In less than the time required for a prey fish to complete a full tail beat, the Barracuda launches forward. Water shears along its narrow frame. The caudal fin, deeply forked and built for thrust, drives explosive acceleration. The predator's body remains rigid, minimizing drag, channeling force directly backward. Prey, reaction time becomes the battlefield. Most small reef fish can detect sudden motion within milliseconds. Their survival depends on it. But the Barracuda compresses the interval between detection and impact so dramatically that avoidance fails. By the time the first sardine registers a looming shadow, the predator is already inside the school. The strike is surgical. Jaws open just enough. The mouth is lined with long, dagger-like teeth, designed not for crushing, but for slicing. The Barracuda does not clamp and wrestle. It slashes. A sideways snap severs muscle tissue, often cutting prey cleanly in half. The predator rarely stops moving. Momentum carries it through the school and out the other side. It then turns. This is where coordination meets limitation. Fast twitch muscle fibers burn through stored energy rapidly. Anaerobic metabolism dominates during the burst, producing lactic acid within muscle tissue. The Barracuda cannot sustain this velocity. After several seconds, power output declines sharply. The predator must either secure prey quickly or abandon the attempt. This biological constraint shapes its hunting strategy. Barracuda rarely engage in prolonged pursuit. Instead, they patrol likely feeding corridors where prey density is high. Reef edges, tidal channels, and drop-offs become ambush highways. The predator positions itself where fish must pass, conserving energy between attacks. But speed alone is not enough. Precision is critical. During acceleration, visual processing must remain stable. The Barracuda's eyes are large, oriented forward enough to allow binocular overlap. This improves depth perception, crucial when closing distance at high speed. A miscalculated strike could mean wasted energy or injury from collision with reef structure. The inner ear and lateral line system provide additional stabilization. Vibrations transmitted through water inform the predator of prey movement patterns. In dense schools, microcurrents created by coordinated swimming become readable signatures. The Barracuda does not simply aim at a single fish. It anticipates collective motion. There is a darker efficiency embedded in this strategy. When the predator tears through a school, the chaos it generates becomes a secondary weapon. Prey scatter unpredictably, breaking formation. Isolated individuals become easier targets. Sometimes the Barracuda does not even consume the first fish it wounds. The panic itself fragments defensive structure. And yet, the Barracuda is not invincible. Its entire survival model hinges on muscle output and reaction time. As it ages, muscle performance declines. Injuries that affect alignment or tail propulsion reduce acceleration. A slight decrease in burst speed can mean the difference between capture and starvation. Temperature also influences its effectiveness. Warmer water increases muscle contraction speed, but reduces dissolved oxygen availability. After repeated bursts, recovery requires oxygen replenishment. The predator must slow down, ventilate, and restore metabolic balance. In cooler water, bursts may be slightly less explosive, but recovery is more efficient. A surprising twist emerges when examining juvenile Barracuda. Young individuals often hunt in loose groups. Coordinated bursts can herd prey against reef structures, amplifying confusion. As they mature, Barracuda become more solitary, relying on individual performance rather than teamwork. Evolution favors independence once muscle output reaches peak efficiency. The reef community recognizes this predator. Smaller fish maintain increased vigilance in zones where Barracuda patrol regularly. Some species alter schooling patterns, tightening formation or staying closer to coral cover. The presence of a single high-speed predator reshapes spatial behavior across entire reef margins. But perhaps the most revealing aspect of Barracuda survival lies in restraint. It does not strike at every opportunity. It calculates energy expenditure implicitly through instinct. If prey density is too low, the predator holds position. If currents are unfavorable, it drifts to more strategic ground. Every burst must justify its metabolic cost. In open water, the barracuda's silhouette glints like polished steel. It appears calm, almost indifferent, but within its muscle fibers, potential energy waits. The slightest trigger, a sudden flash of scales, a deviation in schooling rhythm, ignites the sequence. Acceleration, impact, silence, then recovery. The ocean around it returns to apparent normalcy, but the balance has shifted. One fewer fish, one more burst recorded in muscle memory. In ecosystems where hesitation means hunger and delay means escape, the barracuda lives on the razor edge of reaction time. Its strategy is brutally simple, yet biologically extreme. Push muscle fibers to their limit. Compress the window of prey awareness. End the encounter before fatigue sets in. Survival here is not about patience or camouflage. It is about overwhelming physics. As sunlight fades into deeper blue and the reef transitions toward evening, the barracuda resumes its suspended posture, body aligned with current. It appears almost lazy again, but this stillness is not weakness. It is conservation. Within seconds, it can transform from silver shadow into ballistic force. In a world where prey rely on collective awareness and rapid reflexes, the barracuda survives by being faster than perception itself. Its life is measured in bursts, each one a gamble against exhaustion. The limit is muscle output. The currency is reaction time. And in the thin margin between detection and impact, the barracuda rules. A humpback whale can starve while swimming through oceans that appear endlessly alive. It is one of the largest predators on the planet, a titan of muscle and bone moving through blue immensity. Yet for months at a time, it does not eat. It sings, it mates, it migrates thousands of kilometers across entire ocean basins, burning through the energy of its own body, like a furnace running without fuel input. Every tail stroke is powered by stored reserves. Every breath at the surface is a reminder that mass alone does not guarantee survival. In polar feeding grounds, summer explodes into productivity. Cold waters churn with nutrients drawn upward from the deep. Phytoplankton bloom in dense green clouds, capturing sunlight and converting it into chemical energy. Zooplankton respond. Krill swarm in vast concentrations, each shrimp like crustacean only a few centimeters long, but together forming living rivers that stretch for kilometers. This is the brief window that determines whether a humpback whale will survive the year. The whale arrives lean from migration, its body long and streamlined, its pleated throat grooves folded flat against its underside. Beneath its skin lies a thick layer of blubber. But by the time it reaches feeding grounds, those fat reserves are depleted from travel and breeding effort. What follows is not casual foraging. It is a campaign of intense accumulation. Humpback whales employ a hunting technique known as lunge feeding, a biomechanical feat that pushes the limits of muscle coordination and structural stress. When a dense krill patch is located, often detected through visual cues or subtle changes in water density, the whale accelerates from below. Its massive body surges upward with surprising speed. Mouth closed, pectoral fins stabilizing its trajectory. At the last moment, the jaws swing open to nearly 90 degrees, and the throat pleats expand like an enormous parachute. In a fraction of a second, the whale engulfs a volume of water that can exceed its own body mass. The mouth becomes a cavern filled with seawater and prey. This maneuver imposes tremendous drag forces. Water resistance spikes. The whale's forward momentum drops abruptly as the engulfed mass pushes against its expanding throat. The structural elements of the skull and lower jaw must withstand this hydraulic load without failure. Elastic tissue in the ventral pleats allows the throat to balloon outward, increasing volume dramatically. Specialized sensory organs along the jaw help coordinate timing, ensuring that the mouth closes before structural limits are exceeded. Once the engulfment is complete, the whale clamps its jaws shut and begins expelling water through baleen plates, keratin fringes hanging from the upper jaw. These plates act as filters, trapping krill while water is forced back into the ocean. Each lunge costs energy. Acceleration requires powerful contractions of axial musculature running along the spine. The tail fluke, broad and deeply notched, drives the body forward through vertical strokes. Unlike continuous swimmers such as tuna, humpbacks rely on bursts separated by gliding phases. But the energy expenditure of a single lunge is immense. If prey density is low, the cost can outweigh the gain. This is the central tension of humpback survival. Their size demands enormous caloric intake, but their feeding season is short. In polar waters, daylight persists for long periods during summer, and prey densities peak. The whale may perform dozens of lunges per hour, repeating the cycle again and again. The efficiency of this strategy depends on patch quality. Dense krill aggregations make the physics worthwhile. Sparse patches do not. There is a layer of coordination beyond individual effort. In some regions, humpbacks engage in cooperative feeding known as bubble netting. A group of whales dives beneath a school of fish and begins releasing curtains of bubbles in circular patterns. The rising bubbles form a visual and acoustic barrier, herding fish toward the center. The whales then surge upward through the concentrated prey mass, mouths open wide. This behavior requires timing and spatial awareness. Vocalizations, low-frequency calls transmitted through water, may help synchronize movements. The bubble net is not random. It is constructed with precision, maximizing prey density while minimizing escape routes. Cooperation enhances energy return per lunge, but it also introduces competition. Each whale must position itself effectively to capture sufficient prey. The deeper biological constraint lies in metabolic scaling. A humpback's basal metabolic rate increases with body mass, but not linearly. Larger animals are more efficient per unit mass, yet the absolute energy requirement remains staggering. Blubber accumulation during feeding season must cover not only maintenance costs, but also migration and reproduction. As autumn approaches, prey abundance declines. The whale's body composition changes. Blubber thickens, forming an insulating and energetic reserve. With feeding grounds becoming less productive, the humpback begins its migration toward tropical breeding areas. In these warmer waters, prey density is insufficient to sustain its needs. The whale relies almost entirely on stored fat. Migration distances can span thousands of kilometers. The journey is powered by steady tail beats, each stroke drawing from lipid reserves laid down months earlier. During breeding season, males compete for access to females, investing further energy in displays and combat. Females gestate and later nurse calves using energy derived from blubber stores. Milk is rich in fat, enabling rapid growth of the calf despite the mother's fasting state. The vulnerability of this system becomes apparent when resource availability shifts. Climate variability influences sea ice extent, nutrient upwelling, and plankton blooms. A poor krill year can cascade upward. If prey density drops below threshold levels, whales may not accumulate sufficient reserves. The consequences are delayed but severe. Reduced blubber means lower reproductive success, weakened immunity, and diminished migration endurance. An unexpected twist emerges in how humpbacks locate prey. Beyond visual detection, they may rely on memory of productive areas and subtle environmental cues, such as temperature gradients and salinity boundaries. Oceanographic features like fronts and eddies concentrate plankton, creating predictable feeding zones. The whale's migratory routes often align with these features, suggesting a cognitive map shaped by generational experience. Yet memory cannot compensate for systemic collapse. In regions where krill populations decline due to warming waters or shifting currents, whales must travel farther to locate sufficient food. Increased search time elevates energy expenditure. The margin between surplus and deficit narrows. Despite their immense size, humpbacks are finely balanced machines. Their skeletal structure supports lunging forces. Their circulatory system delivers oxygen to massive muscle groups during repeated dives. Their lungs collapse safely under pressure at depth, preventing nitrogen absorption that would cause decompression sickness. Every system is tuned to maximize feeding efficiency during a narrow seasonal window. As winter approaches in feeding grounds, a final frenzy unfolds. Whales lunge with urgency, stacking energy into blubber. The ocean surface ripples with their movements. Then, gradually, they depart. The polar waters grow quiet, plankton densities wane, and the whales vanish toward warmer seas. Months later, in tropical shallows, a mother and calf rise together for air. The calf's growth is fueled entirely by reserves accumulated in distant, cold oceans. The mother has not fed in weeks. Each breath at the surface represents energy burned, reserves shrinking. The limit of humpback survival is not strength or size. It is the availability of microscopic prey in remote waters. Vast migrations and complex behaviors depend on seasonal pulses of productivity, driven by currents, ice, and sunlight. Remove or diminish that pulse, and the entire life cycle falters. In the feeding grounds of summer, a humpback launches upward once more, jaws opening wide as krill shimmer in dense clouds. The ocean seems inexhaustible. But beneath the spectacle lies a strict accounting. Energy in must exceed energy out. Every lunge is a calculation against time and resource. When the whale finally turns south, blubber thick and muscles primed for travel, it carries within its body the memory of plankton blooms and cold currents. Survival at this scale is not about dominance. It is about timing, abundance, and the fragile continuity of food webs stretching from microscopic algae to the largest animals ever to live. A crab can tear off its own arm and keep fighting. The moment the predator's grip tightens, the limb detaches cleanly at a fracture plane built into the joint. No hesitation, no panic. Muscle fibers contract, connective tissue splits along a predetermined seam, and the captured claw is left thrashing in the jaws of a fish, while the rest of the crab rockets backward into the rocks. What looks like catastrophic injury is, in fact, a calculated escape. On a reef shelf or rocky shoreline, crabs live in a world of constant collision. Octopus arms probe crevices with relentless precision. Triggerfish crunch through shells. Moray eels bite blindly into cracks. Shorebirds stab downward with needle-sharp beaks. Even other crabs engage in territorial combat, locking claws in contests that can end with shattered limbs. Damage here is not an exception. It is routine. Yet the crab does not rely solely on armor. It relies on replaceability. The exoskeleton is the first line of defense, a rigid suit of chitin reinforced with calcium salts. It encases the body like a medieval shield, protecting soft internal organs from crushing bites and abrasion. Spines and ridges add structural complexity, making it harder for predators to secure a grip. But once a limb is seized, the armor becomes a liability. If the predator pulls with enough force, the entire body could be extracted. Evolution has built a release mechanism into the crab's anatomy. At the base of each leg lies a specialized plane of weakness known as an autotomy joint. When triggered by specific muscular contractions or external force, the joint fractures along this plane. The limb detaches quickly, minimizing bleeding and preventing internal damage. Muscles clamp shut, sealing vessels. The crab escapes. The sacrificed limb continues to twitch due to residual neural activity. This movement distracts the predator for precious seconds. In those seconds, the crab vanishes into shadow. But escape comes at a cost. A missing claw means reduced feeding efficiency. A lost walking leg disrupts balance and speed. A male missing a large keeler loses leverage in future contests. Physical integrity defines social status and survival probability. Regeneration is the counterbalance. After limb loss, the crab begins rebuilding from within. During the next molting cycle, a small limb bud forms beneath the exoskeleton at the site of detachment. Molting itself is a vulnerable process. The crab must shed its rigid shell to grow, slipping free from the old exoskeleton while its new one remains soft and pliable. In this window, it is defenseless. Within the soft cuticle, the limb bud expands, fueled by proliferating cells and directed growth signals. Over successive molts, the limb increases in size until it matches the original. The regenerated claw may initially appear smaller or differently shaped, but with time it regains functionality. This regenerative capacity allows crabs to survive repeated injury over their lifetime. In densely populated habitats, it is common to encounter individuals with asymmetrical claws, regrowing legs, or scars etched into shell plates. These marks are not signs of weakness. They are records of survival. Energy allocation governs the limits of this system. Regeneration demands resources. Calcium must be mobilized to rebuild shell segments. Protein synthesis accelerates. Nutrient intake must cover not only maintenance and growth, but also reconstruction. In food-scarce environments, regeneration slows. If molting is delayed due to insufficient energy reserves, the limb bud remains undeveloped. There is a deeper physiological mechanism behind this resilience. Crabs possess undifferentiated cells near limb bases capable of re-entering developmental pathways. Hormonal control orchestrates the timing of regeneration, synchronized with molting cycles regulated by steroids. The endocrine system integrates injury detection with growth scheduling. But regeneration is not unlimited. Severe damage to the central body, particularly the carapace covering vital organs, cannot be reversed in the same way. The crab's survival strategy hinges on the idea that peripheral parts are expendable. Core structures are not. On intertidal shores, waves hurl crabs against rocks. Limbs snap under sudden force. Larger crabs dominate smaller ones, often tearing off claws to weaken rivals. In mangrove swamps, mud crabs clash in territorial disputes that leave both combatants scarred. Even mating interactions can involve aggressive grappling. This constant damage environment shapes behavior. Crabs prefer crevices sized precisely to their body width, reducing exposure to large predators. They move sideways, not out of eccentricity, but mechanical advantage, allowing rapid retreat into narrow spaces. Their compound eyes on stalks provide panoramic vision, detecting threats from multiple angles. When an octopus extends an arm into a hole, the crab braces itself, legs anchoring against substrate. If escape is impossible, it may sacrifice a limb rather than surrender its body. The decision is reflexive, encoded in neuromuscular circuitry. A surprising twist emerges when considering predation dynamics. Some predators learn to exploit regeneration cycles. A crab missing multiple limbs is slower, less coordinated, easier to capture. If injuries accumulate faster than regeneration can restore function, mortality rises. The balance between damage frequency and repair speed determines population stability. In certain ecosystems, human harvesting pressures alter this balance. Fishing traps capture crabs repeatedly, sometimes breaking limbs during release. Although individuals may regenerate, repeated trauma combined with environmental stress can exceed regenerative capacity. Physical integrity becomes progressively compromised. There is also sexual selection intertwined with limb regeneration. In species where males possess enlarged claws used in display and combat, losing the dominant claw shifts mating dynamics. A regenerated claw may initially lack the size advantage required to win contests. However, some crabs can regenerate a lost dominant claw into a larger form over time, flipping asymmetry. This plasticity adds another layer to survival strategy. The ecological consequences ripple outward. Crabs are ecosystem engineers. Burrowing species aerate sediment, influencing nutrient cycling. Shoreline crabs regulate algae and detritus. If injury rates rise due to predator imbalance, crab populations may decline, altering habitat structure. In tidal pools at dusk, a crab missing one leg navigates uneven stone with practiced efficiency. Its gait adjusts, redistributing weight across remaining limbs. The missing segment is a clean stump, sealed and stable. Beneath the shell, a new limb is forming, invisible but inevitable. Physical integrity is not defined by permanence in this world. It is defined by recoverability. The limit of this system lies in cumulative damage. If multiple limbs are lost before regeneration cycles complete, locomotion suffers. Feeding efficiency declines. The crab becomes slower, more vulnerable. Regeneration buys time, not invulnerability. Still, in habitats where injury is unavoidable, the ability to shed and rebuild becomes evolutionary gold. Rather than investing exclusively in impenetrable armor, crabs invest in replaceable parts. The exoskeleton cracks. The limb breaks. The body continues. As the tide recedes and predators retreat to deeper water, crabs emerge from hiding. Some bear fresh scars. Others display newly formed appendages, pale and smaller, but functional. Each one carries the story of a recent encounter. In a landscape of claws, teeth, beaks, and crushing jaws, survival is measured not by avoiding damage but by enduring it. The crab's world does not reward perfection. It rewards persistence. A limb lost is a debt that biology intends to repay. Under moonlight reflecting off wet stone, a crab stands at the edge of its burrow, one claw slightly smaller than the other, shell scarred but intact. It moves sideways into the night, not whole, but whole enough. In this arena, integrity is flexible. What matters is that the core survives long enough to grow again. If the kelp stops moving, it dies. Not the plant, the animal. Among the swaying forests of southern Australian coastline, something that appears to be drifting vegetation hesitates for a fraction too long. The current shifts. Sunlight flickers through green fronds. A shadow passes overhead. And the illusion, held together by posture and patience, nearly collapses. The leafy sea dragon survives by refusing to exist. It does not chase. It does not strike with speed. It does not defend itself with venom or armor. It disappears so completely into its environment that survival depends on a single rule: never break the shape of the seaweed. In shallow, temperate reefs, kelp forests rise from the seabed like submerged trees, their long blades reaching toward the surface. Light filters through layers of swaying ribbons. Every frond moves independently, dancing in slow oscillation with surge and tide. Predators patrol these forests with sharp eyes, tuned to detect motion inconsistent with the rhythm of plants. The leafy sea dragon floats among them, an intricate sculpture of appendages shaped like leaves, each one extending from a slender body in elaborate frills. These appendages are not used for propulsion. They do not function as fins. They exist solely to distort the animal's outline. Every filament breaks up the body's true shape, fragmenting it into a convincing imitation of drifting algae. At first glance, it appears ornamental, a fragile curiosity. But this fragility is deception layered over strategy. Unlike many fish that rely on muscular swimming, the leafy sea dragon propels itself with nearly invisible movements. A small dorsal fin along its back oscillates rapidly, generating forward motion. Tiny pectoral fins near the head provide steering. The body remains rigid, minimizing disturbance. The appendages flutter passively, animated by water movement rather than muscle contraction. The key to survival here is coherence with current. Predators in kelp forests, including larger fish, and even certain seabirds that plunge from above, rely heavily on detecting irregular motion. A sudden dart or stiff posture stands out against the uniform sway of vegetation. The sea dragon must match the cadence of the forest perfectly. It tilts slightly when the surge pushes left. It drifts upward when buoyancy shifts. It never snaps or pivots abruptly. The limit is exposure. If seen clearly, it has little defense. There are no spines capable of deterring attack, no speed bursts to escape, no toxins to discourage predation. Its body is lightly armored compared to many fish, but that armor is insufficient against a determined predator. Its jaws are small, tubular, adapted not for combat but for suction feeding on tiny crustaceans. It feeds on copepods and mysid shrimp, microscopic drifters that move among kelp blades. The sea dragon hunts slowly, hovering near concentrations of prey and drawing them into its elongated snout with rapid suction. Each feeding motion must be subtle. A sudden jerk would betray the disguise. The entire physiology is tuned to patience. Its coloration is not static. Shades of green, yellow, and brown vary slightly between individuals, often reflecting the dominant hues of local vegetation. Pigment cells within the skin can adjust tone marginally, enhancing camouflage. But unlike cephalopods, sea dragons do not rapidly change color. Their camouflage is structural rather than dynamic. This creates a vulnerability when environments shift. Kelp forests are sensitive ecosystems. Storms can tear fronds from anchor points. Heat waves can reduce kelp density. When vegetation thins, The background against which the sea dragon hides becomes patchy. An animal built to vanish among dense fronds becomes conspicuous against open water. A surprising twist lies in reproduction. Male leafy sea dragons carry fertilized eggs beneath their tail on a specialized brood patch. Bright pink eggs cling to the underside, exposed to water flow. For weeks, the male must continue his delicate dance of invisibility while bearing a cluster of highly visible embryos. The brood adds mass and color contrast, potentially compromising camouflage. Yet the forest provides concealment if posture remains correct. The male angles his body so eggs align with sunlit kelp nodules. He drifts through thicker vegetation during daylight hours. His survival and that of his offspring depend on not deviating from the script. There is no backup plan. Unlike schooling fish that rely on numbers, the sea dragon is solitary. Unlike armored species that deter through force, it cannot intimidate. Unlike venomous organisms, it cannot punish curiosity. Its strategy is absolute reliance on remaining undetected. This creates a peculiar psychological illusion for observers. When encountered in the wild, sea dragons often seem fearless. They may drift near divers, unperturbed. But this is not boldness. It is commitment to stillness. Sudden flight would reveal them more effectively than patient drifting. The evolutionary trade-off is stark. By investing heavily in camouflage appendages and reduced musculature for speed, the sea dragon sacrifices rapid escape capability. Energy that might power stronger swimming is allocated to maintaining posture and subtle fin oscillations. Its skeleton is rigid, reducing body flexion. Its body plan rejects the typical fish solution of agility. But in kelp forests, agility is not the primary currency. Visual deception is. Predators scanning the forest process thousands of visual stimuli per second. They ignore uniform patterns and focus on anomalies. The sea dragon's appendages create visual noise, breaking its outline into fractal shapes that mimic torn kelp edges. Even its eye movement is subtle, minimizing glint. Yet no camouflage is perfect. Juvenile sea dragons, smaller and less elaborately adorned, face higher predation risk. Their appendages are proportionally shorter. They must grow into their disguise. During early life stages, mortality is high. Those that survive do so by staying deep within dense vegetation, where light penetration is reduced and outlines blur. There is also a behavioral nuance in how sea dragons orient relative to light. When illuminated from above, they angle their bodies to reduce shadow projection onto substrate. Shadows can betray shape even when coloration matches surroundings. The dragon's posture minimizes distinct outlines. Environmental change threatens this equilibrium. Rising ocean temperatures affect kelp distribution. In areas where kelp forests decline due to warming or grazing by sea urchins, habitat fragmentation occurs. Without continuous vegetation, the sea dragon's invisibility becomes compromised. Conservation efforts now focus not only on the animal itself, but on preserving the structural complexity of its habitat. The survival of the leafy sea dragon is inseparable from the survival of kelp forests. At dusk, as currents slow and the forest settles into gentler sway, a sea dragon drifts among blades of algae. From a distance, it is indistinguishable from torn plant matter. Only a faint oscillation of a nearly invisible fin reveals life. A predatory fish glides past, eyes scanning for irregularity. It detects nothing. The sea dragon remains aligned with the current, appendages trembling like leaves in wind. This is survival, stripped to its most fragile principle. Not strength, not speed, not regeneration or toxin. Absence. In ecosystems where every movement is analyzed by hungry eyes, the greatest defense is to remove oneself from perception. The leafy sea dragon does not fight its predators. It denies them a target. As the tide shifts and the kelp forest breathes with the ocean's rhythm, the sea dragon continues its silent mimicry. Its life is measured in moments of perfect stillness, in micro-adjustments to surge and light. Exposure is fatal. Invisibility is existence. And so it survives, not by dominating its environment, but by dissolving into it completely. The ocean is cold enough to kill in minutes, Yet something black and white slices through it, as if the freezing water were harmless. Beneath a skin of drifting ice, where the temperature hovers just below the point at which fresh water would solidify, a penguin plunges into darkness. The sea presses against it with a silent, relentless force, drawing heat from flesh and bone with mathematical precision. In these waters, exposed human skin would burn with cold before sensation vanished entirely. Blood would slow, muscles would stiffen, but the penguin does not shiver. It does not hesitate. It hunts. As it descends, silver air bubbles stream from its feathers in a glittering trail, each bubble a fragment of warmth refusing to surrender. The water is dense, heavy, unforgiving. Every second submerged is a contest between internal heat and external cold. The physics is simple. Heat flows from warm bodies to cold surroundings. In Antarctica's seas, that flow is steep and merciless. Yet inside this compact diver, core temperature remains astonishingly stable. Its heart continues to beat at a rhythm optimized for oxygen conservation, not panic. Its muscles flex with power. Its eyes adjust to blue twilight. The mystery is not how it hunts here. The mystery is how it does not freeze. The answer begins with architecture. A penguin's body is built as a fortress against thermal loss. Beneath its smooth outer contour lies a dense underlayer of down feathers, interlocked so tightly that wind and water struggle to penetrate. Each feather overlaps the next in a scale-like arrangement, forming a continuous barrier that traps a thin layer of air against the skin. Air is a poor conductor of heat, and this trapped layer becomes a buffer zone, a microclimate hugging the body. When the bird dives, water pressure compresses this air, but it does not vanish entirely. Even in the crushing cold, a residual insulating envelope remains, reducing direct contact between skin and seawater. Below the feathers lies another defense, a thick deposit of subcutaneous fat. This blubber-like layer serves as both insulation and energy reserve. Fat conducts heat far more slowly than muscle, creating a thermal gradient between the warm core and the frigid exterior. But it is not merely passive padding. During prolonged fasting periods, when sea ice traps colonies far from open water, these reserves become metabolic currency. The same fat that insulates against cold fuels the internal furnace that maintains life. Yet insulation alone cannot explain survival in near-zero seas. Water extracts heat approximately 20 times faster than air at the same temperature. Even a well-insulated body risks rapid cooling when submerged for extended dives. The deeper secret lies in circulation. Inside a penguin's flippers and feet, arteries and veins run side by side in tightly packed bundles. Warm, arterial blood traveling from the core passes its heat to the cooler venous blood returning from the extremities. By the time arterial blood reaches the flippers, it has already surrendered much of its warmth. The extremities remain just above freezing, sometimes only a few degrees higher than the surrounding water. Meanwhile, the cooled venous blood absorbs heat before re-entering the body's center. This countercurrent heat exchange system ensures that vital organs stay warm while expendable appendages operate on the edge of ice. The effect is astonishing. A penguin's feet can rest on ice without melting it. Their flippers can slice through frigid currents without draining the core of warmth. The body allows peripheral tissues to cool deliberately, reducing the temperature difference between limb and environment. With less difference, less heat escapes. It is a controlled surrender at the edges to protect the center. On land, another strategy unfolds. When Antarctic winds sweep across colonies at speeds capable of erasing footprints in moments, individual insulation is no longer enough. Penguins gather. Hundreds, sometimes thousands, pressed together in tight formations known as huddles. From above, the mass resembles a shifting, breathing organism. At the perimeter, birds face the brunt of the wind, absorbing cold that would otherwise infiltrate the group. Slowly, almost imperceptibly, individuals rotate. Those on the outside edge work their way inward. Those warmed at the center drift outward. It is not random chaos, but dynamic equilibrium. Heat generated by each body accumulates within the huddle, raising the internal temperature significantly above the surrounding air. The colony becomes a communal thermal battery. This behavior is not sentimental cooperation. It is thermodynamic necessity. A solitary penguin in extreme Antarctic wind can lose heat faster than its metabolism can replace it. In a huddle, metabolic costs decrease. Less energy spent on staying warm means more conserved for survival, reproduction, and growth. The cold, once a lethal adversary, becomes manageable through collective geometry. But cold is not only an external threat. It influences reproduction itself. Eggs are laid in conditions that would freeze embryonic life within minutes. Some species solve this by transferring the egg directly onto the tops of their feet, where it rests against a warm brood patch, insulated by a fold of abdominal skin. The egg never touches ice. It is balanced precariously above a frozen continent, warmed solely by parental dedication and metabolic heat. For weeks, sometimes through months of darkness, adults fast while incubating, burning stored fat to keep both themselves and their developing young alive. Stability inside depends on sacrifice outside. Even the shape of a penguin reveals evolutionary compromise shaped by cold. Their bodies are compact and torpedo-like, minimizing surface area relative to volume. A reduced surface area means less opportunity for heat to escape. Wings have evolved into flippers not only for propulsion, but for structural sturdiness against pressure and cold stress. Short tails, tucked necks, and dense musculature all contribute to conserving warmth. Compared to flying birds, which require lightweight frames and exposed appendages, penguins have traded aerial grace for thermal resilience. Yet there is an irony embedded in this design. The same insulation that protects against cold risks overheating during exertion. When chasing prey underwater or moving across sunlit snow, excess heat can accumulate. Penguins possess the ability to regulate blood flow to the skin, increasing circulation when necessary to shed warmth. They can hold flippers away from the body, exposing vascular surfaces to air for cooling. In extreme cases, they pant lightly to release heat through evaporation. The battle is not simply against freezing, it is against imbalance in either direction. Stability demands constant adjustment. Beneath the surface, dives add another layer of complexity. As penguins plunge deeper, hydrostatic pressure compresses air trapped in feathers and lungs. Buoyancy decreases, allowing efficient descent. At certain depths, trapped air may be reduced so much that insulation diminishes. Yet the duration of these dives is limited by oxygen stores. Mussels contain high concentrations of myoglobin, an oxygen-binding protein that enables extended underwater foraging. Heart rate slows during dives, conserving oxygen for critical organs. Blood is shunted selectively, prioritizing brain and heart while reducing flow to less essential tissues. The body becomes a carefully rationed system, balancing oxygen conservation with thermal protection. The cold ocean, once perceived as a simple threat, reveals itself as a sculptor of physiology. It has shaped circulatory networks, feather microstructures, metabolic strategies, and social behaviors. Every adaptation serves one central purpose: maintaining internal constancy while external forces strip heat relentlessly away. Yet the stability achieved is fragile. Climate shifts that alter sea ice patterns disrupt hunting grounds. Warmer waters may reduce prey abundance or change distribution. Ironically, a slight increase in temperature can destabilize a system built for cold precision. Too little ice and breeding platforms vanish. Too much instability and food becomes inaccessible. Penguins are masters of freezing water survival, but their expertise is finely tuned to a narrow environmental window. As twilight settles over the Antarctic horizon, a colony stands against a pale sky. Wind scours the surface, lifting crystals of ice into the air. Within the huddle, bodies press close, feathers interlocking, hearts beating in quiet synchronization. Out at sea, another penguin surfaces from its dive, exhaling a burst of air before slipping back beneath the ice. The ocean continues to pull at every molecule of warmth it can find. The wind continues its assault. The cold does not relent. And yet inside each compact form, life persists at a stable, unwavering temperature. Blood circulates through countercurrent networks. Fat fuels steady metabolism. Feathers trap invisible shields of air. Behavior and biology merge into a single solution. Hold the center steady while the world freezes around you. In a landscape defined by ice and indifference, survival is not loud. It is not dramatic in a single instant. It is the quiet triumph of equilibrium, maintained second by second against a force that never stops pulling heat away.